Okay, good morning, everyone. We would like to welcome everybody. Good morning, good day, good evening, wherever you are. Welcome to the webinar celebrating the 8th International Biotechnology Symposium via Zoom, emanating from the Livestock Biotech Center located at the Philippine Carabao Center, Science City of Muñoz, Nueva Ecija, Philippines. So to start, let us have the invocation, please. Amen. Thank you so much. So welcome to the webinar once again, and we are welcoming all our participants and our partners, the DA Biotech Program Office, the DA Bureau of Agricultural Research, and the PCC. I am Rodora Romero Aldemita, your host and moderator for today. I am the Executive Director of the International Service for the Acquisition of Agri-Biotech Applications, or ISAAA, a nonprofit international organization based at the International Rice Research Institute, Los Baños, Laguna, Philippines. We envision a world where accurate biotech information empowers farmers and consumers to drive food security through innovation. Our mission is to feed the world with knowledge and biotechnology by supporting informative technologies, fostering impactful partnerships, and facilitating the development of science-based policies toward food-secure communities. So, to maximize participation and comprehension, here are the house rules for all of us. So let's show the house rules. Attendees are required to keep your microphones on mute unless invited to speak. We encourage active engagement and participation during the Q&A session. And so please post your question at the Q&A function. And before the end of the webinar, please click the link or scan the QR code, which we will flash at the end of the webinar, and accomplish the evaluation form to receive your certificate via email. I'm happy to tell you that we have sent out the pre-webinar survey, and we have received a lot of responses, and we will gauge your comprehension before and after the webinar. So this 3-hour webinar is a partnership again between the Livestock Biotech Center and ISAAA, and funded by the Philippine Agriculture and Fisheries Biotechnology Program Office. By the end of the webinar, so we hope to have accomplished the following activity objectives. We should have showcased we should have provided information on cutting-edge biotech innovations that enhances productivity and efficiency, improves animal welfare and health and welfare, and reduces environmental impact. We should have understood how these advancements align with market demands and consumer preferences, facilitating interdisciplinary collaboration among academia, industry, and policymakers. Please take note that this webinar is open to all, so we have various stakeholders represented in this webinar. And so we also have highlighted, we should have highlighted the regulatory frameworks, have highlighted also successful case studies, explored funding opportunities, and developed actionable recommendations to drive the adoption and integration of biotechnology in livestock production. Okay, so to start the ball rolling, let us hear the welcome remarks from Livestock Biotech Center Chief, Dr. Marvin Villanueva. Marvin?
Thank you very much, Dr. Aldimita. So good morning, good afternoon, and good evening to our esteemed guests, distinguished speakers, fellow researchers, policymakers, and industry partners who are joining us on-site and online. It is with immense honor and enthusiasm that I welcome you to the 8th International Livestock Biotechnology Symposium. Today, we convene under the theme Demand-Driven Biotech Innovations, gearing towards a resilient and sustainable livestock industry. This theme embodies our shared commitment to address some of the most pressing issues that the livestock industry is experiencing globally, such as sustainability, climate adaptation, food production, and the ever-evolving market demands. Biotechnology stands as a beacon of transformative potential, unlocking ways and solutions that increase productivity while preserving the environment and uplifting the well-being of countless people whose lives rely on livestock. During this symposium, we will learn about the standing of livestock biotechnology on the international and local levels, unfold the latest advancements in biotechnology, exchange ideas, and build partnerships that will transform the livestock sector into a dynamic and sustainable industry. I would appreciate your contribution and active participation as we join hands to develop science-based, practical, and equitable solutions to the concerns facing society. I urge you to embrace this opportunity to share your perspectives, learn from each other, and build connections that reach far beyond today. I believe that the connections and plans devised here will make a deep impact on our industry and our communities internationally and locally. Once again, I extend my warm welcome to everyone, and I look forward to all the fruitful discussion, and I'm confident that we will create a truly transformative experience through this webinar. Thank you very much.
Thank you so much, Dr. Marvin. Now let's listen to the opening remarks from the Department of Agriculture Biotech Program Office Director, Dr. Paul Limson. Paul?
Thank you so much for the warm introduction. Allow me first to congratulate the DA Livestock Biotech Center and ISAAA for spearheading this multi-stakeholder initiative. Organizing activities like this is vital, especially as we navigate the rapid pace of technological and scientific advancements. Over the last 2 decades, the DA Biotech Program Office has passionately championed the growth of biotechnology here in the Philippines. It hasn't been an easy path. The program started back in the 2000s amid challenging negative perceptions about biotechnology and without any regulatory framework or policies to manage biotechnological products in our environment. Now, we stand at a pivotal moment where biotechnology has become an integral component of our agricultural landscape. Just last year, the DA Livestock Biotech Center successfully held its first regional Livestock Biotech Symposium in Batangas. It was a significant achievement, offering a platform for stakeholders to come together to discuss future directions in livestock biotechnology. Our livestock sector faces a myriad of challenges today, from feeding a growing population to addressing the impacts of climate change things are getting more complex day by day. We have been noticing how extreme weather conditions like droughts and floods impact not only our crops, but also our livestock operations. Given these challenges, now is the ideal time to implement innovative solutions to enhance both the productivity and sustainability of livestock. I am confident that there are opportunities and hope ahead. We have the potential to not only increase productivity, but also make our livestock systems more resilient and efficient than ever before. This year's theme, Demand-Driven Biotech Innovations, Gearing Towards a Resilient and Sustainable Livestock Industry, truly encapsulates the essence of what our community stands for. It is about coming together to create a livestock industry that is both resilient and sustainable. It reflects our collective goal to harness Pinoy biotech advancements to genuinely improve our practices and make a tangible difference. Currently, we have diagnostic kits and optimized protocols at our disposal, ready for our researchers and regulatory offices to use and explore. A portable handheld diagnostic device for quickly spotting key poultry pathogens, a lamp-based protocol for detecting Salmonella enterica in meat. These kinds of advancements are the fruition of years of dedicated research. Sustaining the progress made in biotechnology over the years continues to be a significant challenge. Misinformation about our technology still undermines the potential of biotechnology. However, if there is one thing this symposium aims to communicate, it's this. Research and innovation should not be confined to the academic and industrial sectors alone, but should extend across all areas to benefit generations to come. There is a need to bridge the gap between scientific advancements and real-world applications. We must ensure this innovation reach the grassroots level, empowering local farmers and producers to utilize these technologies effectively. As such, I truly hope we can keep these kinds of discussion going in the coming years. Maraming salamat po at magandang umaga sa ating lahat.
Wow, thank you so much, Dr. Paul Limson. We have heard the challenges that we are facing right now, and we think that animal biotechnology would contribute in solving these problems. And Dr. Paul Limson and Dr. Marvin Villanueva has laid out the fact that in the Philippines, we are gearing up and we want to be at par with a global animal biotech scenario. So thank you so much. Now we move to our first speaker. Our first speaker is a distinguished professor of cooperative extension in the field of animal genomics and biotechnology in the Department of Animal Science at University of California, Davis, where she has been on faculty for over 20 years. She holds a PhD in genetics from UC Davis, and her research focuses on the use of animal genomics and biotechnology in livestock production systems that include work on the uses of DNA information and biotechnologies in beef cattle production systems and the development of genome editing approaches for cattle and sheep. She is a frequently invited resource person, a well-renowned scientist, and has received numerous recognition from the American Association of the Advancement of Science, from the Council, she is a Council for Agriculture, Science, and Technology awardee of the 2014 Borlaug Communication Award, and the American Society of Animal Science 2019 Rockefeller Prentice Award in Animal Breeding and Genetics, to name a few. So colleagues, let us welcome Dr. Alison van Inenam.
Okay, let me just go ahead and share my screen here. Make sure I've got that on. Share from beginning. All right, and all of you can just give me a thumbs up that you can see my screen and hear my voice.
Yes, we can.
All right,
Both.
perfect. All right. Well, hello, everybody, and thank you so much for the invitation. I am thrilled that livestock is the topic of your discussion. It's been my lifelong career to work on the genetic improvement of animals, and often in these biotechnology discussions, livestock don't get a mention, and they are very important. And so we want to make sure that we do include them. So a lot of what I will discuss today is the result of a survey we did of what people are doing with genome editing in livestock. And so biotechnology is much broader than just manipulating an animal's genome, things like vaccines, as was discussed earlier, and tests for whether an animal is sick or not. The focus of my discussion today is going to be specifically modifying the genome of animals to make them have a particular characteristic we want. And so I do want to acknowledge this is a subset of biotechnology, and biotechnology in general is broader than what I'm going to discuss today. And I do want to acknowledge the postdoc that was involved with a lot of the work, Alba Ledesma, that I'm going to be presenting here today. So I do want to start, and I always start with this slide because I think it makes us re-examine or have a bit of a think about the fact that animal breeders can make very dramatic changes to our animal populations based on conventional selection. That is, selecting parents that have the characteristics we want to produce offspring to carry on a particular characteristic. And so these dog breeds have been developed by a group of breeders who thought that, you know, a poodle was a good dog or a dachshund was a good dog. And that's all working on the naturally occurring genetic variation that makes up our animal species. And so in the same way that natural selection and evolution works on naturally occurring genetic variations between human individuals, animal breeders select animals that have a particular appearance based on the background genetic variation that exists, and then select a line that has the characteristics they want. And that's the basis of livestock breeding for our food animals, and has been a very successful component of sustainability, because if you look at a cow today, for example, in California, a dairy cow produces about 4 times more milk than she did in the 1950s. And a lot of that is due to selection for animals that are more productive, more fertile, healthier, long-lived, all of the things that a breeder is wanting to see in their populations. So I want to just put what I'm going to discuss today in a little bit of a time perspective, because genetic engineering, or what's called GMOs, is a technology that's now around about 40 years old, 40. And if we look back, the first transgenic pigs and sheep were produced back in 1985 or so 40 years ago. And there has been some well-known examples. Aqua Advantage salmon, for example, a fast-growing salmon that gets to market weight in half the time, is perhaps familiar to anyone that works in this field. There's Herman, the bull that produced the first genetically engineered product in his that he was able to produce. So he's the first transgenic animal to do a pharmaceutical product, I think lactoferrin, if I remember correctly. And of course, Dolly the sheep, the first cloned sheep. But if we look, there wasn't a lot of products that got to market with conventional genetic engineering. And by that, I mean GMOs, where you're introducing a transgenic construct that's expressing a protein, often from a different species, that gives the plant or animal a characteristic that you want. So in the case of plants, maybe you want it to be insect tolerant. In the case of animals, maybe you want it to get to market weight in half the time. And part of that was due to a very expensive and complicated regulatory process that these types of products have to go through in every country where they've been commercialized. However, about a decade ago, there was a development of a new set of tools called genome editing. And these include technologies like zinc fingers, talons, CRISPR-Cas9 that are slightly different in the way they work to traditional genetic engineering, and which are bringing in a new suite of characteristics that we can incorporate into our animal breeding programs, but also I think kind of putting a new discussion point into regulatory determinations around how best do we, as regulators, manage and regulate genetically edited animals. And so I want to focus today particularly on this last decade of innovation with the editing reagents. And so to make sure we're all on the same page as to what it is we're talking about, I'm going to discuss the fact that genome editing involves the use of a nuclease. And nuclease, by the definition of that word, is basically an enzyme that cuts nucleic acid. And these can be, as I mentioned, zinc fingers, talons, CRISPR-Cas9. They all work in much the same way in that they introduce a double-stranded break to a targeted location in the genome. And that can then be repaired in one of two ways. The first way is what I call this left-hand side of the pathway. So there's a double-stranded break. The enzymes in the cell see that double-stranded break and they try to basically stitch that double-stranded break back together. And when they do that through this process called non-homology end joining, which is a fancy way of saying, let's stick this double-stranded break back together because we don't want our DNA double helix to be broken, you'll sometimes get a small deletion or insertion known as an indel that, if you think about it from the genetic perspective, if you're in a coding region of a gene where each triplet of base pairs will encode for a given amino acid, if you miss one base pair or you add one base pair, you'll throw that entire protein out of frame. And what I mean by that is you'll effectively knock out or inactivate that particular gene. So this pathway, the left-hand pathway, doesn't introduce any novel DNA. It basically inactivates genes. And why would you want to inactivate a gene? Well, maybe that gene produces a protein product that makes your animal susceptible to a disease. And so if you could not have that protein get produced, maybe that renders the animal no longer susceptible to that disease. So that's the SDN1 pathway. This SDN1 is kind of a European term, but it's kind of used throughout the world now to mean I made a double-stranded break, it repaired using the naturally occurring enzymes in the cell, and I didn't provide any kind of a template. So this is exactly analogous to typical genetic variation or mutations, if you will, that we see when we compare genomes of any species. And so if you compare two cows, there's literally going to be millions of genetic variations between them based on naturally occurring genetic variations or mutations. So that's the left-hand pathway. This right-hand pathway is where you provide some sort of a nucleic acid donor template, shown in blue here, that then integrates through something called homology-directed repair to basically repair that double-stranded break in a targeted way. And that can be something as simple as maybe changing a single base pair in where you're targeting that makes it a different amino acid that actually is more favorable to your objective. Or it technically could be used to introduce a gene from another species. So you could technically make a transgenic organism using genome editing. It depends what this donor template is. So this left-hand pathway I'm going to refer to quite a bit. That's the knockout pathway. That is everything that so far has kind of been through a regulatory approval process. The knock-in pathway can exactly mimic a naturally occurring allele that exists in your breed. So for example, you might want to bring in the the allele to not grow horns into dairy cattle, so you don't need to remove their horns mechanically, or you could bring in a protein from a different species and make a transgenic animal. So that's the background of the how, and I'm going to play a little video that hopefully reinforces exactly what I just said. And so without further ado, I will start this.
For centuries, animal breeders have selected individuals displaying desirable qualities and bred them to produce particular goals. Breeders are actually selecting unnaturally occurring DNA sequence variations that result in differences between individuals, like the color of a cow's coat. Using conventional breeding programs, achieving desired changes can take decades, especially in large livestock species like cattle, which have long generation intervals. Innovations in biotechnology are poised to help breeders improve the rate of genetic gain. Genomes are made up of DNA, containing the collection of genes, unique instructions detailing every characteristic of an individual, what they look like, the amount of milk produced, or resistance to disease. Genome editing offers an opportunity to edit these instructions precisely. One method to edit genomes is the CRISPR-Cas9 system. This system has two components: the Cas9 protein, which cuts DNA, and a guide molecule that directs Cas9 where to cut. Bound together, they form a complex that introduces a precise cut in the DNA. The break is then mended by the cell's natural DNA repair machinery. This can be used to inactivate undesired genes, like those that cause disease. Alternatively, the repair machinery can introduce useful DNA sequences which result in desirable traits, like heat tolerance. Genome editing complements traditional breeding methods. It has the potential to introduce useful traits like disease resistance and adaptability into genetic improvement programs, addressing some of the problems facing global agriculture.
If you're interested, that's available on YouTube. It's a good little refresher for people that aren't familiar with what this technology is, and I encourage you to look at it there. So one question that I get asked a lot is, well, how do you go about genome editing a cow? And basically the kind of the thinking is you need to get to the DNA in a cell, and there's kind of two ways that you can make an animal from a cell. The first is that you would work with a newly fertilized embryo. So the sperm has fertilized the egg and produces what's called a zygote, so the one-cell zygote. And what you can do is introduce editing reagents like CRISPR-Cas9 and the guides into that one-cell egg, which is what's shown in this picture. And you can introduce the edits before that embryo starts dividing and dividing and dividing, and then being embryo transferred and put into a cow and producing a calf 9 months later. Alternatively, you could edit a cell and do that same thing and then clone the cell using Dolly somatic cell nuclear transfer cloning methodologies. So the important thing from an animal breeding perspective is that we want to make sure that the edits enter the germline, that is the sperm and the eggs, so that it's passed on to the next generation. Because as animal breeders, if we haven't affected the genetics of that next generation, we haven't done anything. So that's quite different to potentially human medicine, where you're wanting to treat a patient who's maybe got sickle cell anemia and you want to try and cure that patient. We're very directed on trying to introduce these edits into the germline. And ideally, what we would produce is an edited, in this case, cow that has both alleles. So the mum and dad allele would both be edited in a homozygous non-mosaic condition, such that that animal, once it grows up, is going to have both alleles from mum and dad edited. So that's kind of the background, and I'm going to go into a little bit as to how people are doing this editing, or what's the most common approach. Is it embryo editing, shown here? Is it cloning, which would be kind of doing what you do with Dolly, where you basically produce a sheep from a cell, a somatic cell, or, and as the next speaker will cover, the avian world, where they've got an entirely different reproductive system and have to approach things very differently to how mammalian people like me and fish people operate. And so this next slide shows of the peer-reviewed literature that we were able to find working on agricultural applications of genome editing for food production, so not biomedical research, just food production. We found that about 80% of all publications from the peer-reviewed literature use CRISPR-Cas9. So absolutely dwarfs the use of talons and zinc fingers. And these two are older technologies. The concept's the same, so you still get that double-stranded break in the DNA, but the machinery that's used to do it, the scissors, if you will, that cut the DNA are a little bit different, but the concept is the same. So CRISPR-Cas9 very much the driver of current research. And so if we look at the 200 or so applications that we found in the peer-reviewed literature, about 30% were done in cell lines that were followed by cloning. So that's going to be mammals, and that's going to be sheep and cattle and things like that. So 30% through cloning, about 60% through embryo editing. So that's introducing the editing reagents into that newly fertilized zygote, subsequent to the sperm entering the egg. And then the other approaches, the 9% or so, is really where editing is done in primordial germ cells. And I'm going to leave it to my colleague, Mark Tizard, who's going to be discussing poultry editing next, how that's done, because it's a little bit different to how we do it in mammals. But interestingly, the majority of these applications, so about 75 or 3/4 of all of the applications we looked at, were basically knockouts, or that left-hand pathway where we're just inactivating a gene, not introducing any novel DNA. It's just trying to turn off a gene that produces a product that we don't want in our animals. So that's kind of the lay of the land. And then I guess most importantly is the why. Why are we genome editing? And I think it's important to realize that just because we have a new technology or technique, it doesn't change our breeding objectives. We still want healthy, productive, fertile, long-lived, prolific animals. And so our targets haven't changed. We just have a more nuanced way to introduce a useful edit into our elite germplasm, so the very top animals. Maybe we've got a perfect Holstein. One thing we want to change about him is we want to have him not produce horns, for example. Or maybe I've got a perfect pig, but I don't want it to get sick. And so you're going to introduce that using editing into your elite germplasm so that that can then get distributed through the animals that form that breeding population. So what might we want to inactivate or knock out or delete? Basically, genes that are associated with producing allergens, like milk allergens in this case. Maybe we want to alter alleles that make animals more resilient to changing temperatures. So there's an allele called SLIC in cattle that produces animals that are better able to cope with high heat, because they have basically this slick hair coat that basically allows them to keep their body temperature about 1 degree Celsius cooler than their non-slick counterparts. We might want to skew the sex ratio. For example, in pigs, we don't want males because of the boar taint or the unpleasant flavor and odor associated with intact males when they produce pork. So maybe we want to have all female pigs. And that would be something where we only want females, or maybe we don't want the boar taint to develop, so we could inactivate a gene associated with that. We might want to increase yield, so we might do something like a myostatin knockout that results in the double muscled characteristic that is seen in, for example, Belgian blue cattle. Or maybe we want to knock out a part of a gene that makes a pigs susceptible to porcine reproductive and respiratory syndrome virus. So disease susceptibility, increased yield, unwanted development, all things that are part of a normal breeding program, but now we have a more sophisticated way to address it. Similarly, just because we're knocking in a sequence of DNA, or we're using that right-hand pathway where we're providing some DNA template that gives a particular characteristic to animals. We haven't changed our breeding objectives, so we would still like them to be disease resistant or not have disease susceptibility. We don't want unwanted development. We want them to be thermotolerant, improve food, and sex ratio skew, which in the case of female chickens destined for the egg market, we really don't want males because they don't produce a useful product. And I'm sure Mark will address that a little bit in his talk as it relates to layer chickens, where we basically do not want to produce the unproductive sex. So I'll just run through a couple of examples and where this work is going on. So this is work out of China doing a knock-in to produce disease-resistant cattle to tuberculosis. So cattle that are resistant to tuberculosis is one example, a disease-resistant example. Here's an example from the United States where, and this is a really beautiful example, where a breed of cattle called a gyr was edited at a particular location, so a gene called bovine CD46, that the researchers hypothesized if they made an 18 base pair substitution that would alter 6 amino acids, because 18 divided by 3 is 6 amino acids in this particular protein, it would render a protein that still does all of the functions that that protein normally does in the animal, but it would no longer be susceptible to this major viral pathogen known as bovine viral diarrhea virus. And basically what they did is they edited this, this gene in cell lines. They got this 18 base pair substitution, so they substituted that into the middle of the gene and with this, this hypothetical sequence that's going to produce those 6 amino acids that's going to render those animals no longer susceptible to BVD infection. They then cloned that cell into an enucleated oocyte a la Dolly style somatic cell cloning. They produced a calf that when they compared it to a control animal was had reduced susceptibility to this major viral pathogen that's responsible for basically respiratory disease in bovines. So a beautiful example of a hypothesis, a small change, so 18 base pairs in the 3 billion base pairs that make up the bovine genome was changed, producing a functional protein. The animal was healthy and was no longer susceptible to bovine viral diarrhea. And I think that, you know, as a geneticist, I realize I have my own biases, but if you can produce disease-resistant animals rather than having them get sick and having to get treated with antibiotics or maybe dying, that's bad for the animal, it's bad for the farmer, it's bad for the environment. And so disease resistance to me is a really kind of obvious trait to target with this technology that we have. So here's some work out of New Zealand where basically they were able to edit a gene that's responsible for the black in black and white dairy cattle, Holsteins, and change it to kind of a pale brown, with the hypothesis being that they are going to be less likely to absorb heat that way and so better able to cope with a warming environment. This is an example out of the US from Excelligen where they're introducing the allele of the prolactin receptor gene that results in this slick characteristic. So this animal has the slick hair coat as compared to a control, and so the one on the left is able to keep its body temperature 1 degree cooler than the one on the right, and obviously would be better adapted to tropical conditions or hot conditions like in South America or North Australia. Here's an example of a knockout of an major milk allergen. So beta-lactoglobulin is why many people are allergic to milk. And basically this group in New Zealand was able to inactivate that gene so that that cow no longer produced that allergen. So if you are allergic to that protein, you would technically be able to drink that milk because it would no longer contain that protein that results in that allergen. This is work, again, out of the US by Excelligen, where they basically introduced an existing allele that exists in some breeds of cattle like Angus that doesn't grow horns. It's a dominant allele. They were able to introduce it into dairy cattle genetics to produce naturally occurring polled or hornless bulls that were then able to pass that not growing horns or hornless trait onto their offspring. So that's an animal welfare issue in some countries. And this is work that's being done again with Excelogen, trying to develop dairy cattle for subtropical and tropical zones. And they've kind of upped the ante a little bit here and are not only introducing the slick allele, so producing heat-tolerant animals that will produce dairy in tropical zones. They've also introduced 2 other edits that they hypothesize will make these animals resistant to trypanosomiasis, or African sleeping sickness. So this is a multiple edit for heat-adapted, disease-resistant animals that are well adapted to the tropics, and they're working with a base genetics here that's been developed in Brazil that is also tropically adapted. And so those animals are being produced basically to address that subtropical dairy market. And then finally, I'll finish with this example of PRRS, which is porcine reproductive and respiratory syndrome virus. And this is a nasty virus that's endemic in all the areas shown in orange on this map. And it results in a respiratory virus similar to what we've just been through with COVID, but it also results in stillbirths and abortions. And basically, if it hits a pig facility, it's very contagious and it can take out the whole facility. So a devastating disease that has been addressed using genome editing with work originally started at University of Missouri, is also working with the Roslin Institute, and there are groups in China working on this. And what they found was if doing a small deletion in a gene results in pigs that no longer have the access site for that virus to gain access to the pigs. So they're disease-resistant pigs. And as I said earlier, what's important when we're talking about animal breeding is we really need to bring those edits into what's called the nucleus herd, or the elite genetics, the seed stock herd, that is going to then produce the animals that are going into commercial production. And so in the case of pigs, for those of you not familiar with pig breeding, it's a 4-way cross between sire lines and dam lines. So basically, the company that's trying to do this, called Genus, has described a strategy where they're introducing the edits into all 4 of these grandparental lines in the homozygous condition. So they're inactivating the gene in both maternal and paternal alleles in all 4 of these lines. And they've now done this such that when these animals are mated together to produce the commercial pigs, that pig is going to inherit the mutated form of the allele in both from mum and dad, such that it will pass on that disease resistance to its offspring. And so you have to think of it a little bit like plant breeding, but in this case, I guess these are the seeds, if you want. And so we need to edit all of the lines that make up the commercial product in order for it to get passed down. So that gives you some idea of what's being targeted. And this summary kind of shows the traits that are being targeted. So what's shown here in blue is ruminants, so blue, and ruminants are sheep and cattle, goats, things with 4 stomachs, or a rumen. Monogastric animals in purple, that's mostly pigs, but it can also include some other species. Aquatic animals are shown in orange here, avian are shown in yellow, and insects are shown in red. And so what you'll notice is that all of these different varieties of animal classes are being targeted. Yield is number 1 most targeted trait. Reproduction is second. And by reproduction, I mean things like altering the sex ratio or making infertile animals so they can't interbreed with native populations and potentially bring their genetics into a native population. Biotic stress is disease resistant, so the ability to be resistant to disease, a lot of effort there. Then hypoallergenic is product quality, so removing allergens, for example. Multiple would be things like that dairy bull I described, who's not only disease resistant but also heat tolerant. Color is really important in fish, so goldfish there's a reason you put the color in front of a fish, so that's an important one. Quality traits, and then abiotic stress is things like animal welfare, things like not growing horns, for example. So that most of the effort is going into, these traits that are very big profit drivers, but also, and I think importantly, biotic stress. We lose about 20% of our animal production to disease. So if we can use this to help reduce disease, that would be a great thing. So I will just touch on kind of the commercialization aspects now, and I'm going to later on talk in depth about the regulatory aspects, but I do want to bring up the fact that there are actually products already on the market, and that is particularly in the case of Japan, where there are 3 different species of fish that have been genome edited to inactivate a gene that results in them getting to market weight in less time. So they grow faster, for want of a better expression. So these are growth traits, and these fish are able to get to market weight similar to the AquAdvantage salmon, but in this case, we're inactivating a gene, whether it's the leptin receptor or the myostatin gene that we see in other species like the Belgian blue cow. So that's some pictures of them, and these have been developed by an academic scientist who decided to commercialize these fish, and they have actually gone through a regulatory process in Japan and been allowed to go to market, and consumers can purchase these fish. And interestingly, it was expected that there would be quite a lot of pushback because GMOs, or traditional genetic engineering in Japan, has been met with a lot of skepticism and, I would argue, misinformation around the attributes of genetically engineered or GMO plants. And so it was anticipated that bringing these products to the Japanese market would encounter similar resistance. However, what happened was that back in the 90s, when GMOs were first coming out, there was a lot of major news companies and consumer groups that really were questioning the food safety risks and really pushing kind of a fear-based narrative around these technologies. And it was expected that they would employ similar strategies when it comes to gene editing. But interestingly, what happened was that they actually found that rather than people focusing on risks, which is kind of what historically people have been concerned about, having zero risk basically of products coming to market, that there was some attempt to kind of demonize these products and create fear around it, but that just didn't develop into a mass mobilization. And so the media coverage was mostly positive. I think in part because it was a Japanese researcher, an academic, not a big, large multinational corporation, who said, hey, I made these fish. I think they'd be cool to commercialize, and they did. And so it just basically didn't generate the same public outrage that GMOs did. And so it's only one country, but I think it's an interesting example, because if you're expecting there might be pushback, that would be a country I thought we might see that in, and it just hasn't eventuated at this stage. So that's what's going on in Japan. And I think more generally, if you're interested in where in the world is genome editing research going on, I've collaborated with ISAAA and ISAAA to basically develop a gene-edited animal database that's available at the ISAAA website at the link that's shown at the top of this slide. And what we have in this database is basically all of the peer-reviewed papers that I've been discussing in this presentation are listed. And so here, for example, if we go in this database and we look at the different species, we can see Atlantic salmon, sea bream, etc. And so because I'm Australian, I'm going to pick on sheep. So if I look at all the research that's been done on sheep, I can bring up this table. And what this table shows is the year the publication came out, which species we're working on, so in this case it's all sheep, and where that research is being done, what the gene is that they're being targeted. If you're interested in looking at the paper, you can click on the view details. What pops out right away is that a lot of this research is being done in China. And more generally, I think that was a trend that we absolutely saw in our review. And I will just notice that this undergone regulatory process, no button here, means that these are research animals that have been reported in the peer-reviewed literature. They're not been gone through a regulatory process, and they're not commercially available to consumers. So I just want to be clear, this is research, not development. But this trend of a lot of the work being done in China is kind of reinforced by this graph, which shows, again, the different animal categories. And you can see that over half of the 195 or so publications that we looked at were being published by China, with the United States second, Japan coming in third, and you can see a smattering of other countries here in terms of where this work is going on and which different species are being worked on. And so there's a pretty good portfolio here in China. They're pretty much working in all of the different groups of animals. And so if you're more a visual learner, there's more than 100 of the papers, of the 195 papers, were coming out of Chinese labs or had Chinese first authors, was basically how we made the determination, with US coming in second and a smattering of other countries after that. So the other thing, if you go to that gene-edited animal database and you click it to be undergone regulatory process, yes. So if you remember the earlier one, it was no. So that was the research animals. These are ones that have gone through a regulatory process somewhere in the world that's made a determination around them. And so if you want, it's somewhat analogous to the GMO plant approval database, but I'm not going to use the word approval because these are not necessarily being evaluated and approved as a GMO. They're often being evaluated as a conventionally bred product. And I'll get into more details in that when I start talking about the regulations. So what this shows is that in 2018, Argentina made a positive determination for myostatin knockout Nile tilapia, saying that in this case, they are not GMO and they technically could go to market in Argentina if that company wanted them to. Doesn't necessarily mean they have. It just means a regulatory determination has been made. And so if you're interested, you can click on the details and it will take you to the regulatory determination documents from that country and what material and information they considered when giving that regulatory determination for those animals. If that's too small for you to read, this is basically a summary of everything that has got a regulatory determination. So there's really only, 5 countries that have made determinations at this stage. South America is big into this, and you can see, for example, the PRRS-resistant pig that I mentioned earlier, the knockout has been given a determination in Colombia and also in Brazil that came out last year. You can see Argentina these are yield, slick mutation, that's the heat tolerant cow. Myostatin again is a yield one. And you can come down to Japan. There's the 3 fish species that I mentioned earlier that have had determinations. And then finally in the US, there's been what's called enforcement discretion for the slick mutation, that is the heat tolerant mutation in cattle. So that is where we are in terms of current products that have reached the market or been given a positive determination that allows them to be sold if the company so desires. So that's kind of, and as I mentioned, these 3 fish products are actually being sold in Japan and are available to consumers. So hopefully I've given you an overview, and to me, I see editing as a cherry on top of an ice cream sundae. So if I'm an animal breeder, I need to get together with breed association people that agree with me on what a good cow looks like, or a good dog, or a good chicken. We'll develop our breeding goals. That is, what are our objectives? We're going to record in detail all of the characteristics of those animals, how fast they grow, how many eggs they produce, how quickly they rebreed, all of that kind of thing. That's performance recording. We'll do progeny testing to get genetic merit estimates on the bulls. In the case of cattle, we might be doing AI and ET, embryo transfer, genomic selection. And then what we can do is introduce a useful allele, like not growing horns or disease resistance, into this beautiful genetically adapted animal that is our breeding program's outcome. And so it's the cherry on top. It adds a useful trait, but we're working within the existing animal breeding infrastructure. And so whether we're able to do that or not is, in my opinion, very dependent on regulations. And so in summary, genome editing offers an approach to introduce useful genetic variation without the linkage drag that would be associated with crossbreeding. Scaling useful edits to commercial livestock populations is going to be technically complicated, like that pig example I gave, where you're going to have to introduce it into all 4 grandparental lines. In many countries, simple edits or the left-hand pathway, the SDN1, where we're not using any DNA template, are not treated any differently to conventional breeding because there is no foreign DNA. It's non-GMO, if you will. And to me, the fate of this technology in livestock breeding programs is very much dependent on developing a risk-based regulatory framework that is kind of shared amongst countries that trade animal products, and that's pretty much the world. And so with that, I will just acknowledge the groups that fund my lab and also the people that work in my lab, and Alba particularly, who did the review that I basically took all of the information from. So with that, I'll stop sharing, and I'm happy to take questions or however Dr. Aldemita would like to proceed.
Thank you so much, Alison. This is very this is really a very comprehensive presentation of what's going on in animal biotechnology, especially livestock, cattle and sheep and pig and others. And we know that a lot of things are still going to be done as we go along. We are experiencing more enthusiasm from animal biotechnologies, so we will be having more of this products in the very near future. Thank you so much. We will have the Q&A after every presentation. Okay, can you wait a while? Thank you so much.
Sure. Yep.
Right, thank you. So at this point, we would like to invite everyone to put your questions in the Q&A function, and we will be looking at them later after all the presentations. And so we would like now to go to the next presenter, and he is the principal. Oh, let's give Alison a round of applause first, a virtual round of applause. Okay, thanks. Thanks once again. So we'll have the next presentation is from our principal research scientist, Genome Engineering, Health and Biosecurity at CSIRO. He is part of the team that explored the emerging field of RNAi and microRNA biology. His group was the first to catalog the microRNA repertoire of chicken, and improvements in these techniques from his lab have led to very efficient methods to edit the chicken genome. And one spin-off from that is a new method to remove males from the egg layer industry without having to hatch and cull day-old chicks. So to know more about that, let us now welcome Dr. Mark Tizard, and he is a specialist from CSIRO. Mark, are you there now?
I am indeed. Hopefully you can hear me.
Yes, we can.
And
thank you.
I'm aiming to request the share. It's not allowing me to share at the moment.
Can you allow Dr. Mark to share his slides?
Yes, Doc.
Oh, okay. Apologies. I'll quickly say that I'm speaking to you from Australia, a land never ceded by the Indigenous people, and I pay my respects to the Indigenous people here, the land from which I speak, the Wadawurrung people of the Kulin Nation. And I'm continuing to have issues here sharing. This top one. Let's go with sharing that. No. Still not letting me share. Oh, I may have to turn. Oh, hang on. Sorry. Let's try this. Yeah, let's see if we can share
okay.
now.
Joel, can you try and
hang
share
on.
it?
Here we go. Here we go.
Okay.
Right. Share now. How we
right.
go?
Okay. Wonderful.
Are we sharing?
Yeah. Yes.
And you should be seeing the full screen.
Right. Okay.
Yep. All good.
Yes, please.
Excellent.
Continue.
So, yep. I'm always in awe of speaking after Alison. She's done such a great job of walking through what animal biotechnology is. I will also be focusing specifically on things that allow us to change the genome of the animal for a purpose, and in this case for improving agriculture. and food production. So without further ado, what's so good about poultry? And I hope you can see here that poultry is a growing source of animal food. It now, between eggs and poultry meat, matches the production from beef and milk and dairy worldwide for production of high value food for people. I've set the boundaries on this from 1962 to 2023. Data's coming from Our World in Data, a fantastic resource if any of you want to go and have a look at it. And 1962 was when I was born. So this is the improvement. Poultry has become a really important source of food, of high quality animal protein. And it does that in a really, really efficient way in terms of the amount of food that is taken in by a chicken that is then turned into food that we eat from that chicken. And it's bettered by milk, but not by anything else other than aquaculture, fish, which is still a growing space. And in terms of the land use per gram of protein, it is clearly one of the best ways of producing food. And that's important for feeding a growing world population and doing that with minimized impact on the environment. Now, Alison talked about traditional breeding and the goals that breeders have, and that yield is one of the most important. This set of graphs basically shows you the improvement in production per animal of chicken meat and eggs. And I've got in the world and Australia in this one, and the United States in that if you interrogate this source of data, you can pull up whatever country you're most interested in. It's a great resource. But the improvement in chicken meat yield, and again, I've set the date since 1962 when I was born to today, and that's 90% improvement in meat yield from chickens and a 40% improvement in egg yield from egg layers. So why would we use animal biotechnology? Why genetically engineer birds? Well, they've been involved in science and biology for a long time as model systems for developmental biology, and you'll see why in a minute when I show you some pictures. They're also a favorite target for biopharma because the laying of eggs is a great externalization of material, biological material from an animal that can be a high value biological product, a medicine, an antibody, or whatever. And that's matched in the cow space by milk as a great externalized material from which you can extract high values. And conservation, we've been working in looking at how to modify birds and save endangered species. And that's work that's going on around the world. But agriculture is really our focus today, and improved poultry production, meat and eggs is obviously a key thing. As I've said, they're already really good, but we know we can do better. One of the areas is around disease resilience. And I know that many of you will be thoroughly familiar with avian influenza or bird flu, but there are many other diseases that impact the production of chickens and both meat and eggs. And that's something that is a lurking pandemic again for the world. And it's a horrible term to use, but it is out there. There's a current outbreak of H5N1, which has shown itself capable of jumping from birds into mammals and from mammals into humans. And it's quite scary. So that's a critical area, and one in which selective breeding is really quite difficult to target. Sex sorting for the layer industry is another area that is practically impossible to affect by using the standard selective breeding methods that have improved production in general. And safer food products, eggs in particular, that have allergens that can be quite dangerous for some people and very upsetting and have to be excluded from diets for many people, for particularly young people who have these allergies, but many that don't grow out of them. So these are things, again, that are really difficult to address by standard breeding methods. So I had to jump in and quickly adjust my slides. So I'll let you all get these new slides since Alison threw to me a very good point, which is that gene editing in birds is very different from gene editing in mammals. So in mammals, you can get at that single cell zygote stage, the fertilized ovum that you can edit and adjust or replace the nucleus with an edited nucleus and then breed up a whole animal. That's not straightforward, but it's very doable with mammals. But with birds, their reproductive system is so significantly different that that really isn't possible. The ovum is intimately associated with the yolk that we're all familiar with in eggs. And the yolk is produced off a little track of developing yolks that come around the ovary and that are released around about one a day into the top end of the reproductive tract. And then it's coated in protein, in a membrane, and then ultimately at the bottom end in a shell before it's passed out into the world where we find it. To be able to do this kind of editing means extracting a yolk with an ovum on the surface, modifying that, and then putting it back. And that was tried many, many years ago, and it is really unsuccessful. So how do we do it? We do it by a very different method, and that is to get at the early embryo to attack, if you like, to access the germline. So this is how we do it. The developing embryo that you saw in that last picture, and we'll come back to, has a circulating blood system. And in that circulating blood system, not only are the white blood cells and the red blood cells, but what we call primordial germ cells. And these are cells that will go on to populate the gonad, the reproductive organ that generates the gametes, the egg and the sperm. And they're circulating in the bloodstream, and we can inject into that bloodstream constructs of DNA and that may be CRISPR, or it may be a technique that we use, which is a transposon. And those bits of DNA get inside the cells, and they'll get into different cells. But the ones that we're most interested in are these, the primordial germ cells. And so where it's gone into primordial germ cells, great. Where it's gone into other cells, it's kind of irrelevant, but it will happen. Now, those primordial germ cells will ultimately, during the early development of the embryo, populate what's called the gonadal ridge, which becomes the reproductive organ, the gonad, which is either the testis or the ovary. And so here are the cells coming in and populating, and some of those cells have been modified. What happens is they grow, and there's a cluster of cells comes from that growth, and individual cells that have taken up DNA will be modified. And they'll be modified potentially in different positions when as we do use a transposon. But if it's CRISPR, it will be a very specific target in the genome. So this is how we do it. We basically open up an egg at about 2 days of development after incubation. So the fertilized egg has a little ball of cells, and you'll see that later as well. As it grows in 2 days, you see a structure like this, the embryo in the middle and the blood vessels either side that gather nutrients and the oxygen that it needs to breathe, if you like, and to grow. And we inject, and here's a needle injecting into the dorsal aorta. We then seal the egg back up. So we've cracked a little hole in the eggshell to see this, and then we seal that hole back up, and then we incubate and we hatch. We hatch eggs to release both hens and roosters, but we're interested in the roosters because they have a large number of gametes that they'll release, i.e. sperm, when we analyze them, and we're looking for a few sperm that will have been changed. We do that by PCR, and when we find an animal that has a positive signal by PCR in its sperm, we breed from that animal, and then we hatch chicks from that. And so this is the process. About 50 eggs, about 25 of those will hatch. We grow those to maturity, and about 10 of those will be males. We select from the males, and then we start breeding from those males. And in this case, we would have selected about 50 hatchlings. And ultimately, what we'll see if we're getting eggs through that are transgenic is those hatchlings, when they grow up, if they're female, will be carrying a transgene in our case, but could just be an edit. If it's an edit, it's a little bit more sophisticated to identify this. If we add in a marker gene with the transposon system that we use, that marker gene may be green or red, and we end up with a bird that either fully carries throughout its body the green gene or the red gene. Now, we've been doing this for many years in our search for things that can interfere with influenza virus reproduction, to block the growth of bird flu in the bird, to keep that bird protected. But along the way, we realized that when a bird inherits this, it inherits a single copy, and that single copy is either in one or other of the offspring. And if those offspring are male or female, then you get the marker with either one of those sexes. But you can be better than that. You can actually place it on the chromosome that determines sex. And so that's one of the things that we ended up targeting. It's an ethical dilemma and potentially a welfare issue for the egg-laying industry. And that is very simple. As you will all know, hens lay eggs, roosters don't. And the genetics of chickens has been improved so much, just as we have with cattle and beef and dairy. You don't eat steaks from a dairy cow and you don't drink milk from a beef cow. Their genetics are different. They're expert at one or the other. And the same with chickens. They're either really good at making meat or they're really good at laying eggs. And unfortunately, on the egg laying side, the males that are hatched, their body is telling them, don't make muscle, turn that into an egg, but they've got no egg to make. So they're really poor in production characteristics. Their meat isn't good, and the yield in terms of how much food you have to feed them to get meat protein back, and the duration that you have to keep them for are all wrong. Farmers won't farm them. It's not a sensible way forward. So the end result in Australia is 20 million male chicks are culled every year. That's 300 million male chicks culled in the US every year, and 7 billion male chicks culled globally every year. The industry really wants and indeed needs a solution to this. It's not great, and I'm not going to show you the means of culling because it's not the best thing in the world. But it's inescapable at the moment. So the current global practice is incubating the eggs for 21 days in a nice warm incubator, 37 degrees, and then hatching everything, manually sexing those eggs, and then removing and culling, manually sexing the chicks that hatch, and removing and culling the male chicks. In Europe, they've been working, and I'll explain why in a minute, with technologies to try and get around this. They're fairly direct technologies, and they're a great improvement, but they require incubation up to around about day 9, and then sampling from those eggs, and then analysis and reassortment. So analysis to find out which ones are male and female, and you can tell that at this early stage. And then reassorting them to remove the male eggs and to just put back the female eggs that are then incubated, but you're removing and culling an embryo. So that egg is now partially used up by the development of the embryo. What we've been aiming at is using markers, the markers that you saw before, to do marker sorting at point of lay. And as you saw at point of lay, there's a little ball of cells, and I'll come back to that one again, that will carry the marker and that will glow either red or green. And red is the selected color because it's more straightforward to detect. This is basically detection and removal at point of lay. So no incubation, no growth, and no culling of the males. And that's actually pretty significant. When you think there's 7 billion male chicks culled every year, that's because there's 7 billion hens laying eggs around the world. But 14 billion eggs had to go into incubators. That's a big incubator there. That's a lot of carbon footprint in production. So there's an issue. It's being tackled. There are big improvements that have been achieved in Europe, but there's a potential complete solution. These are a couple of the things that have been going on in Europe. So Respect uses a laser system to blast a little hole in the shell, a needle that goes in and takes a sample, and there's a test for the sex hormones. Very similar in OVO in Holland. So this is in Germany. In Holland, OVO has developed a system that does something very similar. It uses gas chromatography rather than an ELISA that's used here to look for products of the sex hormones. And they work really well, but they have to incubate all eggs. They have to take those eggs out, grab a sample, do an analysis, reassort the eggs, and then put them back. So what was driving this development in Europe was countries that have actually banned chick culling. So in France and Germany, it is now illegal to cull male chicks. In Italy, it's coming, and it's being considered in many countries around the world because it's a practice the industry wants to move away from. So there are a whole raft of new technologies that were driven by the imperative to get around this. And Here are some of the technologies that are now in place: Cheggy, Select, which I showed you, Inova, which I showed you, and Orbum. They're all various methodologies that are either invasive or non-invasive, and that means taking a sample or not taking a sample. Not taking a sample is obviously preferable, but comes with its own problems. And none of these use biotechnology. They're all means of doing it, but they all require growth out to at least 8 to 10 days at the end of the day. So the alternatives that are actually in development, CSIRO, another company called Exit that's using the same system as we are, and Next10, which uses a different system. Now, CSIRO and Exit, we're basically running side by side. We think we may have the advantage here, but it's point of lay and it's using that marker system. It's not invasive, but it does use biotechnology. Next10 uses a male-linked terminator gene, which is a really clever system. I take my hats off to them, which actually uses a light system to activate a gene in only the male embryos, which causes them to stop growing. But they have to incubate all the eggs. So there's a distinction in this system. Now, all of these are still in very early stages of development. We don't know how developed Exit is with theirs. They come out of Israel. Next10 comes out of Israel as well. But I'll tell you a little bit in a minute about how we're progressing with ours. But biotechnology, so that's it's a question mark. Is this going to be acceptable or not? So why is the biotechnology needed? Because the legislation in France and Germany is there banning male chick culling, and that is going to spread, we know. Sampling analysis systems reduce the hatchability as well, and they involve complex machinery, incur additional costs, and increase the carbon footprint, which you really want to try and go the other way if possible. But if you can stop actually incubating and hatching, there are sustainability benefits in that. So stopping prior to incubation, which is really good and also presents the value to be able to recover high value protein from the eggs that you don't put into the incubator. There's really great egg white and egg yolk there for possible recovery. Reducing the carbon footprint. If you halve the number of eggs, and that's in the millions, then you're halving the amount of space and time and effort and energy going into incubating those eggs. Reduce carbon footprint, very, very attractive. And improving the ethics and welfare aspects of egg production. Welfare is always a question that's asked about this, and that's really about, is the culling done appropriately? There are rules and regulations about how the animals should be culled, but sadly, those are not always followed. The ethics is the key thing, bringing to life these little chicks and then snuffing out that life without the animal having use. Now, in agriculture and animal agriculture, we all know the end point is that the animals will sacrifice themselves, or we sacrifice them to recover the food value from them. But in this case, there is no food value in those male chicks, but there could be from the male egg if it's removed. How does it work? Well, it's based on the segregation of the sex chromosomes. We all know that in humans, XY, that's all the guys that are listening into this presentation, and the ladies listening into the presentation, XX. So it's the male sperm that determines the offspring's sex. The offspring of your kids, if you have them, will have been determined by the male. In chickens, it's back to front. So the female is actually heterogametic. She has a Z and a W chromosome. The male has only ZZ. So it's her ovum that determines the outcome. And so this is the animal that we need to get to with genome engineering, to put a marker, in this case the red marker, on her Z chromosome, and she will pass that on only to the male offspring that she generates. And therefore, we can identify and remove that egg at point of lay and only incubate and hatch the hens. And this is what it looks like. This is a blastoderm under red light with an appropriate filter showing you the red fluorescent protein, in this case, actually on the surface of the yolk, and that little ball of cells that you can barely see there, that is the blastoderm, about 60,000 cells at the point of lay of an egg. How it would work would be hens laying these eggs would lay a whole mix of eggs, male and female, that carry or don't carry the marker. A detection system, which is in development right now, will actually pull out the presence of the marker and separate those out. That becomes a tray full of male eggs that are not incubated, and a tray full of female eggs that then go off to be incubated. They hatch and they become the layer hens that generate the food eggs that we eat. On the male side, well, at the moment, the objective is this carries a transgene. It's a GM material. And so right now, it's like, well, they'll be removed completely and taken out of the system. But there are industrial and potentially food values to be gained from those eggs. And that's something that's under exploration. How does this work? Now, this is equivalent to the diagram that Alison showed you before. Chickens and pigs have something in common, apart from being actually pretty good at producing food and pretty good at doing that off a small footprint of land. But they all use 4 genetic lines to ultimately breed together traits from the 4 parent lines or pure lines that are brought together in the final production bird, the commercial layer in this case. And so what we have to do and are beginning to explore, and I'll talk through that in a brief moment, is to put the marker gene on both copies of the Z of the pure line in the male and the single copy of the Z in the female in the pure line of the C line. And the reason being that the breeding process going down leads to a ZZ rooster in the grandparent that will breed with the D line and give you a CD cross that is female and that carries the fluorescent marker on her Z chromosome. And so when she mates with an ABZ, the Z goes with the Z here. ZZ is the male, and that's the one that's removed from the system. The female that comes from this, the ZAB and the Z and the WCD, come to make a ZW female, the hen, and she'll go to the hen house and she'll make eggs that become food products for us. Now that pyramid points down to a single animal, but in real life, the numbers go the opposite way. There are very small numbers of pedigree birds that actually will be carrying the marker. They're bred through grandparent and great-grandparent stock to the parent stock that the female carries the Z on the marker on her Z chromosome. And then she gives only the W to her hens, so the commercial layers. And so there's an event called null segregation, and that is the segregation of the Z and W chromosomes when ovum are formed, when the determination of male or female happens, and the male gets it and the female doesn't. And that's a null event in the segregation of the sex chromosome. So the female doesn't have it. She is a null segregant, and that's done by point of lay detection. So from commercial layers down to the consumer, there's no marker present anymore, no GM material. So we've spoken to, and this is just for Australia, but we've got maps like this for a range of countries around the world. We've spoken to everyone that has a little green star, the global genetics companies, the national breeders, the egg producers, the supermarkets, which is really important because you have to have an outlet for this, and consumers and egg buyers. We've spoken with animal welfare groups who are very supportive of this, and equipment companies, but most importantly, the regulators, the people who formulate policy and those who regulate foods that are using biotechnology. And that's the Office of the Gene Technology Regulator and Food Standards Australia, New Zealand. So this is how the thing would look ultimately, with a global genetics company involved, get the marker in. The national breeders and hatchers would use a detection system to separate that out, and the marker is removed with no male culling. The hens going to the hen house, and the eggs on the shelf, and the eggs for the consumer, nothing changes. They're the same hens that will go into the hen house. Same eggs to the supermarket or to the food product producer, same egg products that everyone will eat. No marker from this point onwards. And that you get improved genetics, improved welfare and ethics, improved sustainability, and a reduced carbon footprint. So will this be regulated as a GM food? We've spoken with the Office of the Gene Technology Regulator here in Australia, and recent amendments in 2019 to the regulations, the gene technology regulations, say that organisms derived from GMOs but with no traits from gene technology are non-GMO. And so offspring have not inherited the trait, are non-GMO. So what about the regulators more broadly? Are they happy with this? Well, it's based on null segregant processes that are used widely and accepted in plant breeding. They're happy with it in principle. Food Standards Australia New Zealand has said that basically a null segregant is likely only to require equivalence testing because the food product is essentially the same as it is today. Of course, both OGTR and FSANZ will oversee this when we produce the eggs finally, and that is, I guess, underway right now. Final decision will come when we can show them data. But everyone that we've spoken to agrees that this looks like it will get the green light. We've done some consultation globally, and we've gone out and spoken with people from Cannabia in Argentina, CTN Bio in Brazil, Health Canada, in Japan, the National Agriculture the Food Research Organization, and the USDA, FDA, and the Center for Veterinary Medicine. And we're getting green lights, question marks in all of these, but green lights from all of them. None of them can see a reason why under the null segregation process there would be an objection, but the final decision rests on presentation of appropriate data packages. And that's something that I guess Alison may talk about later in terms of approaches to the regulators, which is really a critical stage in all of these things moving forward. So remember, this is GMO at an early stage in development, but the hen that comes from this is not a GMO, and neither is the egg that's produced as food. So this is an interesting scenario. I think that this is an opportunity to fly things with the public where animal biotechnology is used, but where they're not faced with a product where they have to look at it and go, Now, I don't want to get wrong with this, because I think that the GM product itself would be fine. So if this marker was still present, or if there was residue from it still present, there is nothing to be concerned about in that. However, in this case, it isn't, and so it's a way of starting the conversation. Now, are we doing this? The answer is yes. We are now working with one of the global genetics giants, that's Hendrix Genetics. They've established a partnership with us to look at an innovative sex sorting technology for the layer industry, and it shows that the global genetics companies are really taking the topic of removing culling of male chicks really seriously. They're getting into this, and they're really working in really great partnerships with us. Lastly, our genome engineering team. So a group of us here, myself, Olivier Seralbo, Kirsten Morris, Xuning Shi, and Terry O'Neill, who's our team leader, and Terry Wise, who's not in the photograph but was heavily involved in this, and Caitlin Cooper, who used to work with us, have all been really critical in getting this moving forward. We have a really great supporting team in the vet, Ali Kaneen, and teams at our Werribee animal facility and a small animal facility. We have animal welfare officer involvement all through this, because they want to be sure that there's no adverse effects, and that's a key factor for the global genetics partners, that there should be no adverse effect from any of the stuff that we do here. Just quickly shout out Tim Doran, who helped to establish this whole team and the initiatives and open doors for opportunities that we've had. Finally, just to say thank you again to the organisers. Thank you all for listening. I'll be happy to take questions as well. And I know this is a bit English speaker oriented, but if you ever wondered what comes first, the chicken or the egg, in the English dictionary, it's very clearly the chicken. So thank you.
Wow. That one made me laugh. Okay, we haven't really sorted that one, which comes the egg or the chicken, but now you said it's the chicken. Thank you so much, Mark. And that gave us an overview of what's happening with the null segregants in chicken. And we know that in poultry, there are still other researchers that will deals with animal welfare and animal health. I mean, poultry health and poultry welfare. I know there might be questions on that, but thank you so much, Mark. We'll have you again later with the questions. So let's give Dr. Mark Tizard, a big round of applause. Thank you so much. And so we go to now the regulatory aspect, and this will be provided to us by Dr. Alison Van Inenam once again. Thank you.
Oops. Nope. Sorry, I am just making sure I share here.
Okay.
Are you seeing my slides?
Yes.
Okay, I'm still seeing Mark's. I'm not sure why.
Ah.
Or I'm seeing welcome to the Livestock Biotech Center.
I see the global regulatory landscape. Would you like to unshare and then share again?
No. So long as you guys are seeing it, I'll just proceed.
Can you move?
Proceed.
All right.
Yeah.
Sure, please.
Well, hang on. Nope, that's disappeared from my screen. So I am going to unshare and share again, I think. Stop share.
Yeah.
Share. Okay, let me. Okay, are you seeing that now?
Yes.
Okay.
Right.
I'm not sure exactly what's going on, but I am going to be giving this presentation on behalf of my colleague, Diane Ray Crane, who is with the USDA, who was not able to present this afternoon. And so I do it a little bit with trepidation because this is not technically my area of expertise. And if anything that's incorrect is my fault, and anything that sounds good was Diane's doing. So we'll just leave it at that. So, I think that what is the role of regulations is undoubtedly to protect health and safety of humans, animals, and the environment. That's the ultimate goal. Theoretically, to help instill trust in the food supply. And more importantly, or I guess what I find most compelling, is the fact that we have to have a good regulatory process that enables safe, useful innovations to get to market. And that tension between absolute safety and allowing products to market, I think exists. And there's got to be a balance between risks and benefits, because a lot of these technologies have tremendous benefits. They address very real problems like animal disease or single-sex chickens. And not allowing safe technologies to come to market is also associated with risks, and that is the risks of doing nothing, basically, to address problems.
Yeah.
So different countries have different regulatory approaches because different countries have different rules and regulations, and it's not possible for all countries to have the exact same regulation. And that's because of the laws and their enabling authorities differ. And they may have different regulatory triggers. So some may be product-based, like in Canada, or some may be based on definitions to do with the technology, like whether they're a GMO or not. That's a process-based law. And many countries chose to put into place specific laws to regulate genetically modified organisms, or GMOs, or genetically engineered organisms 40 years ago when they were first introduced in plants and animals. Others chose to use existing laws, like the United States. Canada used a product novelty approach. But in general, I would say they all determined, with GMO products, where you've got the expression of a new protein, that's the transgene, there was a general agreement that you needed to do a food safety evaluation. And that was kind of set up under the Codex guidelines. And that's basically an international group under the FAO, World Health Organization, that has a task force to determine, well, what would you do? How would you do a risk analysis of food derived from modern biotechnology? And particularly of relevance to this discussion is the 2008 guidance that came out on the conduct of food safety assessments for foods from recombinant DNA animals. So by the wording there, you can see they were anticipating genetically engineered animals with a recombinant DNA construct, that is a transgene that they're expressing, and how would you go about doing food safety assessments for that? But things have changed since then, because basically the Codex evaluates the nature of the rDNA construct and its expression. Well, given the background I've given you tonight, if you're a gene-edited animal that doesn't have any novel foreign DNA, there is no recombinant DNA construct or expression product. You just have a genetic change that maybe stops an endogenous protein getting expressed. So that discussion around the food safety aspects of the expression product kind of goes away, because there isn't an expression product. The health status of the animal, so how healthy is the animal, and whether the composition of the food products from that animal differs from conventionally bred animals. That's kind of the main things that are considered. And that has been put in place because that was what we were dealing with, recombinant DNA animals. Notably, the guideline doesn't address animal welfare, ethical, moral, or socioeconomic aspects. It's or environmental risks. It's about the safety of the product itself. So with the evaluation or the evolution of genome editing, where we've got something different to a recombinant DNA animal, we've got an animal whose native genome has been slightly altered to give it a characteristic, we kind of need to rethink this regulatory landscape. And so there's an effort in many countries to modernize their regulatory approaches. The goal hasn't changed. We still want safe food, whether it's conventional or biotech. And so those laws are in place for conventionally bred animals too. You are not allowed to sell an unsafe product. It doesn't matter what breeding method you use to produce that product. So that's always the top priority. But new regulatory approaches are really focusing more on characteristics and potential risk of the products rather than the process used to produce them, and to encourage the creation of innovative, safe products to address global challenges such as disease resistance, climate change, population growth, etc. And so how can we actually facilitate using these new breeding tools for farmers within current production systems? And I think both Mark and I gave very clear examples in the pig and the poultry industry where this technology is getting introgressed or introduced into existing elite genetics. That is the four-way cross that makes up the broiler chickens, or the layer chickens that Mark showed, or the four-way cross that makes up the pigs. And that's really the priority and the importance of getting safe innovation to farmers. This is perhaps the most important slide, I think, in my opinion, and I love this slide because it really summarizes it down. So the question that regulatory officials have been asking is, when do we regulate a product under the GMO guidelines, and when do we treat it as just conventional breeding, which doesn't have this added layer of regulatory evaluation? And I think that there's general agreement that natural mutations, so the single nucleotide polymorphisms and the genetic variation that differentiates a Holstein from an Angus cow or a broiler chicken from a layer chicken, is not going to be regulated as a GMO. So yes, there's genetic changes between them, but it was naturally occurring and it's not treated any differently. And let's face it, animal breeding has a history of very safe use. So I think there's also agreement that if you actually introduce a transgenic recombinant DNA insertion, then that's going to be regulated as a GMO, because that was kind of the definition of a GMO. So it's carrying extra recombinant DNA that's producing a product. The focus of the debate really is the categories in the middle, which are kind of shown in yellow here. And these are the changes possible with genome editing. And so I think I mentioned that kind of the left-hand pathway would be this no template, small changes, deletions. The right-hand pathway is kind of providing this template, which can be short and just maybe change a single base pair, or it can be a long template that's maybe an allele from one breed going into a different breed. And really, it's where to draw this line is where the debates are happening. And that is where many countries have drawn the line above this, this latter group down here. So basically, if you're using a repair template that's transgenic, that is going to create a GMO in many countries. If you're using a repair template that exists within the genetics of that species, then it's cisgenic, or it's a short template or small alterations. That's not going to be treated as a GMO. And really, it's this foreign DNA switch that is determining whether or not this is going to be treated as a GMO or not. And a little bit, basically above the green line, you're conventional. Below the green dash line, you're going to be regulated as a GMO. And that's partly derived from the definition of the Cartagena Protocol, where foreign DNA is a really important trigger. And in the case of the ones above the green dash line here, there is no foreign DNA. There's potentially DNA from a different breed, but you could mate an Angus to a Holstein and get an animal that doesn't grow horns. You just bring along all that beef genetics that you don't want in your elite dairy cattle germplasm, in the same way you wouldn't want to cross a broiler chicken with a layer chicken and dilute the excellence of those two specific lines. So the definition of a and really, to be honest, it's this smudgy bit here is where it gets really not so obvious in different regions as to whether or not it's going to be treated as a GMO or not. So some places are treating long repair templates or cisgenes as GMO, and others it has to be this foreign DNA trigger to be a GMO. And as I mentioned, it's really the definition of the LMO and the Cartagena protocols. So in 2015, Argentina became the first country to publish a new approach for the regulation of products created using technologies. So they looked at the Cartagena Protocol and its definition of a living modified organism, or LMO, and they said, okay, it has to have a novel combination of genetic materials obtained through the use of biotechnology. And modern biotechnology is defined as using recombinant DNA and direct injection of nucleic acids into cells or organelles. But importantly, it's the combination of the two that makes you a GMO. So you have to have not only the use of modern biotechnology, but you have to have a novel combination of genetic material. And that's been interpreted to be foreign DNA. And that combination, if both of those are not true, then it's not considered to be a GMO, basically. And that has basically spread to this regulatory map that I'm showing here, which is saying that in many countries, they're putting into place a regulatory landscape where in the green shown here, you have regulatory exclusions. And typically that exclusion is for animals that don't contain any foreign DNA. It does depend a little bit on the regulation definition, but that's kind of how it's come about. Some countries are still developing their regulations. Some don't have any regulations yet. And some countries, for example, here, South Africa, is treating all genome edited animals and plants as GMOs, whether they're just a knockout or whether they actually have foreign DNA. So irrespective, they're kind of just saying you used modern biotechnology, therefore it's a GMO. And so if we break this down a little bit, we can kind of go through. So this map evolves daily. I think I have the most recent map here, and if I don't, I apologize, but Diane did get it to me. And so basically we have Argentina, Brazil, and Chile kind of following in Argentina's lead, and basically saying you have to have foreign DNA to be regulated as a GMO. Otherwise, you're going to be treated as conventional breeding. And basically the Latin American countries have all kind of gone down that pathway. Canada has a novel foods approach. So if you've got a novel product that hasn't been in the food supply before, you have to go through regulations irrespective of the technology used to produce that product. The United States is using the its guidance is to allow some food animals that don't contain foreign DNA to be brought to market through enforcement discretion. And then we've got a whole range of different outcomes in different countries. So Israel's put in place policies with exemptions for genome edited plants, but haven't yet made a determination on animals. Nigeria, Kenya, and a growing number of African countries are adopting policies that are generally in alignment with the kind of the approach that was spearheaded by Argentina. Japan is similar, so very similar to the South American countries, exempting GMO requirements unless there's foreign DNA templates. Australia is a little bit complicated. So at the moment, the left-hand pathway, so deletions are excluded. Whereas if you go down the right-hand pathway with a template, irrespective of the nature of that template, it's a GMO, but that is actually under consideration and revision in Australia. The Philippines, Thailand and Singapore have adopted similar policies for plants, and the animal policies are under consideration. That's kind of part of the whole reason for today's discussion. India and Indonesia have developed draft policies, and China and Korea, albeit with fewer exclusions. And then the UK has created some exclusions for plants and animals and is currently kind of getting its animal regulations under advisement. Norway has proposed a tiered approach, and EU is also considering exclusions. And at the moment, they're kind of still developing their regulations. And really the odd one out is South Africa, where a court ruling determined that genome editing will be regulated under GMO rules. A relatively recent addition to this is New Zealand, who were going down the South African pathway, but have recently announced they plan to update the rules to basically allow simple edits, SDN1's left-hand pathway, to just be treated as conventional breeding. And so, like I say, it's in flux, and this is a very opportune time to be having this meeting. And so when it comes to commercialization, as I alluded to in my presentation earlier, there have been some countries moving forward with a path to allow products to come to market. And so the path for commercialization began with Argentina, who basically made a non-GMO determination of a genome-edited food animal and concluded that, in this case, I think it was a knockout tilapia, was not a GMO, but rather a conventional animal. And then Brazil followed suit in 2019. In 2021, Japan became the first country to commercialize a gene-edited animal for food. And in 2022, the USA made an enforcement discretion decision for a food animal, the slick cattle. And then in 2023, Colombia became the first country to make a non-GMO determination for the PRRS-resistant pig. And I believe, if I'm not mistaken, Brazil followed suit in 2024. So even that handful of applications is more than the 2 genetically engineered animal food products that ever got to market. So that was the Aqua Advantage salmon and the Gelsafe pig. And so what regulations you have in place and whether they allow safe products to come to market really affects what comes to market and what types of problems can be addressed with this technology. And I guess, how regulations are implemented and applied really shapes what products are developed and who can afford to use these technologies. And there was a question in the question and answers earlier around how does genetic engineering affect biodiversity? And I really want to emphasize that I would argue what regulatory approach is selected for biotechnology is how it will affect diversity, because if it is virtually impossible to bring a product to market, but for a large multinational corporation, you're going to have a lot narrower base of genetics getting affected by this technology. So if you compare the South African approach, that is everything that uses modern biotechnology is a GMO and you have to go through full GMO, versus some of the genome edited products are conventional, that is the ones that don't have any foreign DNA, then you get a really different situation. So if it's the no exclusions, a very, very hard line, you just use modern biotechnology, only large multinational corporations are going to be able to afford to bring products to market. And that's basically what we've seen with GMO plants. Developers tend to come from high income countries dominated by large acreage crops with high return. And there was very few food animals. As I said, there were only 2 that were ever approved globally after 40 years, which is a pretty sad record. Whereas if you have the ability to bring edited animals to market, like that researcher in Japan, then you can actually have public researchers and small and medium enterprise companies bringing products to markets. You'll have more countries that are doing approvals. We've already got 5 countries with products that have been given a positive regulatory determination with genome edited animals. You'll start to see the whole technology flourish into different fields like livestock and fruits and vegetables and flowers. And you'll get consumer-oriented traits like, I don't know, the pink pineapple, for example. And most importantly, you'll have more rapid solutions to regional problems. And I back this up with data. So this is from Argentina, where basically when they had the no exclusions approach for GMOs, almost all of their GMO crop approvals were from foreign multinational companies, 90%, with very, very small number coming from local companies and foreign small and medium enterprise companies. When they moved to the approach where they said, okay, we're going to not regulate products that don't contain foreign DNA, what you see is that the majority of the applications are coming from local, that is Argentinian companies and public researchers, so academic scientists like me. Very few, less than 10% from foreign multinationals and 32% from foreign small and medium enterprises. So 90% to 9%. And this to me is really important because when it's coming from large multinational corporations, that's when a lot of the distrust gets sown. And it's like, I don't know that I trust them to do the good for my country versus Mr. Joe Blow, professor I know at my local university, has developed a disease-resistant chicken. I really want to support that because that disease is a problem for me, and I trust that researcher who's doing this work in the country that I work in. And additionally, you get an increased diversity of organisms and traits that are getting targeted. Again, data from Argentina. So when it was only GMOs, you had pretty much everything was plants, a little bit of vaccination work here in microorganisms, but it was pretty much plants and 2 traits, so herbicide tolerance and insect resistance. Important traits, but when you opened it up to have not only that, but novel traits are being regulated as non-GMO, then you start to see a diversity of not only plants, but animals shown here in red, and much more, a bigger range of traits. So not only herbicide and tolerance, but things like health benefits for consumer, added value for industrial use, something that's being developed for consumer preference, like the greens here that don't have a bitter taste to them. So you get a diversity of products that are being brought to market. And I think that data is basically comparing, when they changed their regulatory, what the type of products that were produced and who was producing them, and in a much more, I would argue, democratic way than the GMO debate or the GMO regulations allowed. And so I'll kind of finish with this, 2 more regulatory scenarios. And this gets a little bit back to the discussion around biodiversity. So you can consider genome editing to be something that produces a product that is kind of the status quo. So is a horned dairy cow safe to eat, or are Mark's chickens safe to eat? And the product is the chicken. Or you can say, you know what, this is just a new breeding tool, and it brings up a useful way to bring in traits into our existing breeding programs. And the reason that I bring this contrast is it kind of shows you how treating it as a product differs from treating it as a breeding pool, because how the regulations are applied or implemented impacts other protection goals. So we're not only interested in human safety foremost, but we also would like efficient production systems and sustainable production systems and disease-resistant animals. And that has to be balanced against a regulatory approach that potentially is so expensive and cost prohibitive that only large multinational companies are able to do it. So if we think about the creation of a new product, we're thinking about a genome animal that's created, for example, and then there's a trait it gets introduced and you get this animal and it gets approved as a founder animal. Then the developer would basically multiply that genetics into their breeding herds, and that trait would go into a number of, in this case, Holstein animals. So large companies will supply the gene-edited genetics in Holsteins. But if you know anything about cattle, there's lots of different breeds. And not only Holsteins, we'd also like all of them to be, for example, disease resistant as well. And so if we have to do every single one on a case-by-case basis in each breed, it's going to become very difficult to basically edit all of these different breeds. And so the big breeds will get affected in the same way corn and soy did with GMOs, but the little breeds will not be part of the discussion. And maybe your native indigenous breeds in the Philippines won't be part of the discussion because there's really no way to be able to afford this kind of product-based approach. As distinct from if we actually try regulating this under a new breeding tool approach, regulatory decisions will be made for traits that can be applied to all sexually compatible breeds and species. And therefore, the trait could be freely introduced into other types of animals, allowing, in this case, for the protection of all of the different breeds of cattle. So if we treat it more like a tool and less like the cow is the product, then you have the ability to introduce that tool once it's been evaluated into different breeds, and you can protect your genetic diversity. You'll get a lot more publicly developed traits available to farmers, and you'll be able to be nimble enough to introduce things like flu-resistant chickens if we're able to produce an edit that enables that to happen. And so to me, this is kind of a better approach. And we're really at a regulatory crossroads right now. And I would argue that the status quo of regulating everything as a GMO has not served humanity well. There has not been unique threats or risks associated with this technology, and we've got 40 years of safe use at this stage. Do we need this incredibly high regulatory bar, or is there a way to do it differently? And so I think that countries will need to implement change to ensure the solutions developed are available to farmers, and that's what's not been happening with the existing one. And, you know, we look at a lot of potential risks of approving products. So is the food safe to eat? Is the milk safe to drink? Is the animal healthy? Is there any risk to the environment? And that's all fine and well, but we're not really looking at the countervailing potential benefits. So why are we doing this editing? Well, disease resistance, single-sex chickens, welfare traits, you know, all sorts of things. If we could, for example, get avian flu-resistant chickens, that would be a really big benefit. And what's not really kind of part of the discussion at the moment is what's the risk of not doing anything and just saying, yeah, we've got a solution, but we're not going to use it just because there might be risks. And so I think there needs to be a better risk-benefit analysis, because everything has risks, but not doing something has risks too, especially if the risk is not solving a problem that would otherwise be solved by a safe innovation. And I feel like, you know, this little slide, we've considered the potential risk of everything except the risk of avoiding all risks. And that's kind of the precautionary principle, or basically being frozen from making a decision because you're too risk adverse. And we really do need product-based, science-based, and risk proportionate regulations for genome editing. And I think Argentina led the way in a reasonable approach to ensure safety, but also allow low-risk products to be basically treated in the same way as conventional breeding. So regulatory approaches have to enable safe products to come to market. That's important, and encourage the development of new ideas and innovations. And at the end of the day, we're trying to provide farmers with the choice of the best selection tools to best meet the challenges of their future more sustainably. And I think that's the end of the slides, and I will turn it back over to the moderator to lead us on.
Thank you so much. Actually, I've been laughing and smiling at every slide that you show. It's really an eye-opener for everyone on how contributions of animal biotechnology, crop biotechnology will bring to us and the risks are not really that much. I mean, they can be addressed without harming the potentials of all of these innovations. Thank you so much, Allison and Diane. And so we'll have to go to the last speaker. I know that there are a lot of questions. And to the speakers, if you can see the Q&A, we can start nimbling on how we are going to respond to those. They are very basic questions that needs to be answered. And so before that, let's go to our homemade, how do you call this? A scientist from the University of the Philippines, Los Baños. And she is a virologist with a PhD in biomedical and life sciences from Lancaster University, UK. She specializes in molecular virology, molecular biology, and bioinformatics, and has worked on developing CRISPR-Cas-based vaccines against poultry viruses. She has been a visiting scientist at Iowa State University, and a research scholar at the Boyce Thompson Institute for Plant Research, Cornell University, US. She's committed to improving animal health through research. So let's welcome Dr. Julian Villela. Take it away, Julian.
Thank you. Thank you for that wonderful introduction, Dr. Aldinita. Please allow me to share my slides. Okay, I hope everyone can see my slide.
Yes, we can. Thank you.
Right. Good morning again, everyone. It is a pleasure to be here today to discuss animal biotechnology in the Philippines, its current status, innovation, and regulatory landscape. Thank you again for the organizers for inviting me. And again, I'm Julian, and I specialize in genome editing and viral vector vaccine development. In this talk, I'd like to be able to provide an overview of the current status of animal biotechnology in the country, the Philippines, its ongoing research efforts, regulatory framework that are in place, and how we can actually improve the biotech adoption for sustainable agriculture and livestock health. And then now let's begin by understanding why biotechnology is crucial in the livestock sector in the Philippines. So the livestock industry is a pillar of Philippine agriculture. It has contributed significantly to food security and rural livelihoods. However, disease outbreaks, low productivity, and climate change pose major challenges. Biotechnology offers a cutting-edge solutions such as genome selection, gene editing, and recombinant vaccines and diagnostic tools. Adopting these innovations can transform the industry by improving animal health, boosting productivity, and ensuring a resilient food supply. In the Philippines, there's a lot of research effort with government and academic institutions driving innovation. However, there are a lot of challenges, and these challenges include funding, public perception, and infrastructure. Today, allow me to highlight the progress that we have made throughout the years, regulatory landscapes, and the future of this field. Here at the Institute of Animal Science in UPLB, one of our main efforts has been unlocking genetic blueprint of Philippine native breeds. Through the whole genome sequencing of native animals, we have identified unique genetic variations that make these animals more resilient to heat, diseases, and local environmental conditions. This knowledge allows us to create selective breeding strategies that can improve productivity while preserving genetic diversity. Our work on native cattle and darag chicken, or Philippine native chicken, ensures we harness the best traits for future generations of livestock. Another project that we are very proud of is the genetic variation analysis using GWAS. But why is GWAS very important in this study? Because it was able to help us to connect specific genetic variants to observable traits. In this study, we analyzed the genomes of the darag Philippine native chicken using high-density SNP chips and identified genetic regions associated with higher egg production. This allows the breeders to do selection of superior chickens based on their genetic potential rather than just physical appearance, leading to a faster genetic gains in productivity. And another breakthrough in the Philippine animal biotechnology is, of course, the IDETECH. It's a locally developed nucleic acid detection kit by Dr. Joy Banayo and Ms. Kathleen Manese. This tool ensures that the meat products sold in the market are correctly labeled, preventing food fraud. With this rapid, cost-effective solution, we can safeguard food authenticity, protect the consumers, and improve export potential. Well, animal biotechnology in the Philippines is still in its early stage, but has seen significant progress in key areas. These examples are just fraction of our ongoing work at the Institute of Animal Science, PCC, BAI, and its collaborating institutions. From livestock breeding to disease resistance studies, numerous initiatives are underway to enhance agriculture productivity through biotechnology. Some of the notable developments were led by UPLB, the Philippine Genome Center Agriculture, Institute of Animal Sciences, BioTech, BAI, DALBC, and we have been working a lot to ensure that biotechnology is used for food security and animal health. However, challenges remain. This will include limited funding, lengthy regulatory approval processes, and lack of infrastructure for large-scale biotech application, which eventually hinders the progress. While these genetic and diagnostics tools are very crucial in livestock improvement, one of the most transformative application of biotechnology, as has been described earlier, even in vaccine development, is the emerging demand for a faster, more precise, and adaptable solution for vaccine development. And this is where CRISPR technology comes in. In the next section, let me just discuss with you how CRISPR-Cas9 was used to develop next-generation vaccines that not only protect against existing diseases, but can also be rapidly modified to combat new and evolving viral threats. In the Philippines and worldwide, poultry is a major food source. However, poultry are faced with diseases like avian influenza, infectious bronchitis virus, NDV, which pose very serious threats to food production. And traditional vaccines, however important they are, have limitations, particularly in keeping up with rapidly mutating viruses. CRISPR-based vaccines will provide an innovative solution by allowing us to precisely insert productive genes or protective genes into viral vectors, resulting into a stronger, longer-lasting immunity. Now, as part of a collaboration with the Lancaster University in the United Kingdom, what we did was to develop a recombinant vaccine using this novel technology of CRISPR-Cas9 genome editing against important poultry diseases like Newcastle disease virus and infectious laryngotracheitis virus. So how did we do it? Let's now break down how CRISPR was able to help create new vaccines. So in the beginning, we used Cas9 and guide RNA to cut the viral genome precisely. So the viral genome that we were trying to cut was the viral vector, or the vehicle that will carry the antigen. The viral genome that we use is the ILTV. The next step is after the cut has been made, we will have a foreign gene, such as the NDV proteins or the NDV gene, the Newcastle disease gene, inserted into the viral genome. Now, once we have the recombinant viral vector plus the foreign gene, we will now have a recombinant viral vector that can train the immune system to recognize both our ILTV, which is our vehicle or our viral vector, and the NDV, which is our antigen, providing dual protection. This technique allows for a faster vaccine development and, of course, customization for emerging viral strains. And we love to call it in the lab as the plug and play. So it's a very flexible system that can be used for other livestock diseases. Now, after lab-based validation, we were able to test the vaccine in animals to ensure that it's both safe and effective. Animal trials for this candidate vaccines were done in other Asian countries. However, some of our data are still embargoed. And another thing is that it is imperative that we also do this in the Philippines. We have data from other countries and from other setups, but it is important for us to do it as well here in the Philippines. And this is one of the key research that I've been doing at the moment. In our case, we were able to do preclinical trials, or these are the lab-based validations. Validations to confirm that the putative vaccine behaves as expected in cell models. We were able to do this and replicated it many times. In terms of animal trials, this helped evaluate the immune response of the vaccine, its safety, and its efficacy. It has been done in other countries, but we are still trying to replicate it here in the Philippines. In terms of field trials and commercialization, I know it's still a long way to go, but the regulatory approval is required for a large-scale use, ensuring compliance with the national biosafety guidelines. This CRISPR-based vaccine development, it represents a major leap forward in animal biotechnology here in the Philippines, and I think as well in the world. Unlike traditional vaccines, CRISPR allows us for precise genetic modifications, enabling the creation of a safer, more effective, and rapidly adaptable vaccines against emerging livestock diseases. By using these viral vectors such as ILTV, we can deliver specific traits into we can deliver specific genes into the genome of a recombinant vector. Now, the key question is that, is the Philippine biosafety regulatory framework ready for these advancements? Are you ready? As we explore other applications of CRISPR, such as gene editing in livestock and next-generation therapeutics, we must assess whether our current biosafety guidelines can accommodate these breakthroughs, or if reforms are needed to support innovation while ensuring safety and ethical considerations. So, as explored in our previous discussion, the potential of CRISPR and other gene editing technologies in animal technology is crucial to assess whether our current biosafety regulations are equipped to handle these advancements. The Philippines has an established framework for regulating genetically modified organisms. But gene-edited animals and vaccines represent new challenges, of course, and that requires a specific guidelines. At present, the regulation of genetically modified animals falls under the oversight of multiple agencies, and this includes the National Committee on Biosafety of the Philippines, or the NCBP, the Bureau of Animal Industry, and the Food and Drug Administration. This agency ensures that biotech products are assessed for safety, efficacy, and environmental impact before approval and commercialization. So to address this current innovations, there is a draft of a joint circular on genetically modified animals. The government has introduced this draft for rules and regulations for research and development, handling and use, transboundary movement, release into the environment, and management of genetically modified animals and animal products derived from the use of modern biotechnology. This draft aims to provide a clear regulatory pathway for genetically modified and gene-edited animals, ensuring that developments in CRISPR-based vaccines and gene editing applications are properly evaluated and monitored. So the draft The draft JDC outlines requirements for contained research, field trials, use of international trades of genetically modified animals and products. But there's an exemption wherein gene-edited animals without novel genetic combinations are not covered. And it also seeks to harmonize Philippine regulations and international biosafety standards, facilitating both local innovation and global competitiveness. However, despite this progress, key questions remain. So how will gene-edited animals be classified under this framework? Will CRISPR-derived vaccines, as we've been developing in our lab, and disease-resistant livestock be regulated as GMOs, or will it be treated as a separate category? These are very critical considerations as the Philippines navigate the evolving landscape of animal biotechnology. With CRISPR and other gene editing technologies reshaping animal biotechnology, regulatory frameworks must evolve to keep pace. In the next section, let me just try to discuss what improvements are needed to ensure that the Philippines remains at the forefront of responsible and innovative biotech development. So the CRISPR gene editing technology are transforming animal biotechnology. Of course, we already know that. But are the regulations ready, as we keep on asking, to support this innovation? While the draft JDC is a step forward, more work is needed to ensure clear, efficient, and science-driven regulatory framework. Yeah, I think this image was also used by Dr. Alson earlier. I've also come across this publication. And I think one of the key areas of improvement will always be the clear guidelines for CRISPR in livestock and vaccine. So unlike GMOs, as discussed earlier, CRISPR edits can be precise and DNA-free. Yet, our current regulations here in the Philippines, all biotech application falls under the same lens. Countries like the US differentiate gene-edited animals from GMOs, allowing faster approvals. Should the Philippines do the same? Or should we follow Argentina, for example? Establishing specific guidelines for CRISPR vaccine and livestock will provide clarity for researchers and investors. The second one will be streamlining approval pathways. Right now, as you've seen earlier, biotech approval involved multiple agencies, making the progress a little bit slower and more complicated. So I believe that we need a faster risk-based approvals, especially for CRISPR vaccines addressing high-priority livestock diseases. By differentiating gene-edited products from traditional GMOs, we can speed up innovation without compromising, of course, the safety of the public. The second one, which for me is very important, is public awareness and engagement. So science means nothing if the public doesn't trust it or the public doesn't trust us, researchers. Many people still equate gene editing with GMOs, leading to resistance. Countries like Japan and Canada have tackled this through education and transparency and industry dialogues. I believe we must do the same, engaging farmers, policymakers, and consumers to build confidence in safe, ethical biotechnology solutions. Looking ahead, the US uses risk-based approvals, and the EU has a stricter GMO-like regulations, while Japan and Canada take a more flexible approach, and as well as Argentina. The Philippines must find the right balance, I believe, one that fosters innovation while ensuring safety and ethical responsibility. The future of animal biotechnology, I feel, is here, but regulations must evolve to keep up. We need clearer policies, faster approvals, and stronger public engagement to ensure that the Philippines remains a leader in responsible biotech innovation. Next, let me just try to explore where this technology is headed, this CRISPR technology or gene editing technology. It's heading to disease-resistant livestock, multi-omics research, and next-generation vaccines. So we see here the ethical considerations and public perception. So we see scientific advancements, as I've told you earlier, mean little if the public doesn't trust them or the public doesn't understand it. I think one of the most common concern is that, is gene editing the same as GMO? So I think one of the biggest misconception is equating CRISPR gene editing with traditional GMOs. Because we all know, unlike GMOs, CRISPR that does not always introduce foreign genes or foreign DNA. Instead, it's a very precise, natural-like modification with an organism's genome. However, to the public, these nuances aren't always clear, right? And I think it was also asked earlier in the Q&A, are gene-edited animals safe to eat? So gene-edited livestock, such as the PRRS-resistant pigs developed by the Russell Institute and Genus, had undergone rigorous safety evaluations. However, public concerns about food safety and unintended effects remains. And transparent risk assessment and regulatory oversight are crucial to addressing these fears. And another question I think for me is very important as well is that, is gene editing ethical? Some critics will argue that modifying an animal genome for human benefit raises ethical questions, particularly in welfare, natural integrity, and unintended consequences. While gene editing can improve animal health and reduce diseases, I think it's very important to conduct research responsibly and to engage an open ethical discussions. And we should address misinformation through public outreach, education, and transparent risk assessment. For me, ethical concerns and public perception can either accelerate or hinder biotech progress. Without trust, of course, in everything that we do, even the best technologies will struggle to gain acceptance. Moving forward, transparent communication, responsible research, and clear regulations will be key in shaping the future of CRISPR and gene editing in livestock. So the future direction, animal biotechnology is evolving every minute, as the previous speakers have said earlier. And it's evolving at an unprecedented pace. It's changing every minute. From genome sequencing, GWAS, CRISPR, we've made in the Philippines significant strides. But where is this technology headed next? So the future of animal biotechnology lies greatly in precision editing, disease-resistant livestock, multi-omics integration, and advanced vaccine platforms. This innovation, as I believe, will transform agriculture, making it more sustainable, efficient, and resilient. In terms of gene editing resistant livestock, one of the most promising application, I think, are the disease-resistant livestock, as repeatedly discussed earlier, the PRRS-resistant pigs, which prevents virus from infecting pigs. This approach can be replicated and applied to combat other diseases like ASF, avian influenza, and other livestock diseases. But the big question is, will gene-edited livestock be accepted, or will the consumers accept it? Will the regulatory bodies have the appropriate guidelines to regulate it? And countries like Japan and the US are making progress, but widespread adoption will require clear policies and consumer trust. Another is, we're entering an era of multi-omics. It's the integration now of everything that we've learned, from genomics, transcriptomics, proteomics, and metabolomics. At this point, we were able to gain a little bit of a complete understanding of livestock health, growth, and productivity. And by combining these layers, we can identify key genetic markers for disease resistance, heat tolerance, and feed efficiency. Multi-omics research generates massive data sets. So the challenge now is developing efficient bioinformatics pipeline to analyze and interpret information quickly and accurately. And of course, CRISPR-based vaccines are just the beginning. Scientists are now exploring other types of vaccine development, like RNA vaccines, as the same as the COVID-19 vaccine. And these vaccines could be introduced or produced rapidly and customized for emerging diseases. But the challenge here in the Philippines is scaling up the production or even putting up an infrastructure for vaccine production and ensuring that there is cold chain stability for RNA vaccines in tropical climates like the Philippines. And of course, collaboration between universities, government agencies, and the private sector is very crucial in driving biotechnology innovation. And the challenge in the Philippines is strengthening our intellectual property laws, not only and biotech-friendly policies to attract more private investors in the Philippine animal biotechnology. And the future of animal biotechnology is bright and full of possibilities, from gene-edited resistant livestock to multi-omics and next-generation vaccines. But scientific breakthroughs alone aren't enough. For these technologies to succeed, we need stronger regulations, public trust, and industry collaborations. Now, let me just wrap it up. I also use that image. So let me just wrap up everything. Animal biotechnology in the Philippines is advancing with every innovations that had been introduced. However, scientific progress must be matched by strong regulations, public trust, and continued investment, of course. And as a recommendation, the future of biotechnology or animal biotechnology is here. And the Philippines must take decisive steps to ensure that we are not just adapters of the technology, but leaders in innovation. If we invest in research, strengthen policies, and engage the public, we can transform agriculture and livestock production for the better. And as we look ahead, the question is no longer if biotechnology will shape the future of agriculture. It's how quickly we can adapt and implement its responsibly. Let's continue looking forward to build scientifically progressive, ethically sound, and globally competitive biotechnology industry in the Philippines. Let me just close with gratitude and collaboration to a lot of people who have helped us. Thank you so much.
Thank you so much, Julian, Dr. Julian Villela, a young scientist. She's teaching at the UPLB and a researcher at the same time. Thank you so much. Okay, before we start, our open forum, I would like just to eliminate some of the questions that are there pertaining to the certificates. Everyone who would be responding to the post-webinar survey will immediately receive their certificates in their inboxes, in their email inboxes. And also that we will be posting, we have already permission from our speakers to post the PDF files of their presentation as webinar files at our ISAAA website and also at the Livestock Biotech Center website. And we'd like to call your attention to visit the ISAAA and the UC Davis collaborative project on the gene-edited animal biotech, which are already in place since September last year. So let's call in all our speakers. Can you put us in spotlight, please? And we will have our speakers, Dr. Alison Van Ininam, Dr. Mark Tizart, Dr. Where are you, Julian Villela? How do you put this on the spotlight, please? Joel? Yeah. So put us in the spotlight. That means all four of us in one frame here in the wallpaper. Can you do that? Okay. That's Julian. And then we'll have Dr. Mark Teaser, Dr. Allison, and myself. So while waiting for the spotlighting, I'd like to call the attention of our speakers who have already responded to some of the questions. And for Allison, have you, I know that you have answered some of those questions. Did you find any striking questions that you would like to raise here openly that you have already answered that you think would be an interesting one to raise as well?
Yeah, well, they're all interesting, and so I
Okay.
I didn't answer them to stop having a discussion about them. It was more like there was a lot, and so I wanted to kind of get to them. But just I think one thing that has come up a couple of times is the difference between genetic engineering and gene editing, and I think Julian alluded to that in her presentation as well. And unfortunately, it's not easy to say this is this and this is that because they differ, and so historically, traditionally, genetic engineering or GMOs has utilized a process where a transgene or a genetic construct integrates randomly in the genome of a plant or an animal. You have no control where it goes in. And then the breeders would select for the plants that were herbicide tolerant or whatever trait you were after. And you really had no control over where that transgene went. So that was kind of, it was a one-trick pony to some extent. You could do that. Gene editing is so much more powerful, but also different. So the difference is the precision of the targeting. So the guide RNA, in the case of CRISPR, tells it to go not anywhere in the genome, but to this specific location amongst the 3 billion base pairs in the genome of a cow. You want to go to chromosome 17 at position number 695231 and make a double-stranded cut there. So there's a precision of where it happens. And then there's also not necessarily an integration of a transgenic construct. You can, like Mark's example, but often not. So 75% of the applications we looked at were just the left-hand pathway, so no construct. So all you're doing is tweaking the endogenous or native DNA at a very targeted location. And that is very different to GMOs, but you could technically make a transgenic animal with genome editing. So it's a little bit nuanced and a complicated answer, and one that I get a lot. And I think scientists do a horrible job of using the correct terminology. So I've seen genetic engineering, which is the technically correct term. I've seen that get used to abbreviate gene editing, GE. And it's like, guys, if we're trying to separate these two and we're using the same GE acronym, and genetic modification literally means nothing. It's just an activist term. So, I would argue a Chihuahua is genetically modified relative to a wolf, but it's just through conventional breeding. And so I realize it's a little bit semantic, these distinctions, but, for scientists, language is important and precision is important. And I think people that haven't been involved in this field for 30 years maybe just use, GE, who cares? We'll just abbreviate it to gene editing. It can be GE. And anyone that's worked in this space is like, no, please don't do that, because we don't want to get tied up in the whole GMO debate, because it is a different technology. And, the example Julian gave of that beautiful virus or vaccine development using CRISPR-Cas9, like that's very different to what you can do with genetic engineering. And so I think it's unfortunate, but and certainly groups that are opposed to this technology want that conflated. They want that confusion, because if they can get the GMO fear into people around gene editing, that's going to work to their advantage. So I think it's important to be very precise with our language, even though it's frustrating. And it is important to kind of say where they're alike and where they differ.
Correct. Thank you so much. And then the issue of safety, we have questions on can we eat the GMs, or can we eat gene edited animals?
Well, that's like asking, is it safe to eat mushrooms?
Yes. Okay.
Often, but not always, right? Depends on the mushroom. So I think that, gene editing, genetic engineering, they're both tools to produce something. And depending what you produce and what you're expressing in the case of a GMO will depend on the safety of the product. And so you can't, it's like saying, is electricity safe? Well, not if you're in an electric chair, but it is when I'm sitting here on Zoom, I'm not worried about getting electrocuted. So I think it has to be a more nuanced question. It's a case-by-case basis of what animal, what trait, or what gene, or did you inactivate or turn on or express, because that product is what is going to be safe or unsafe, not whether you used genome editing or genetic engineering. And so it's a case-by-case basis, and there's no such thing as just a blanket, everything produced using electricity is safe, because clearly it's not if you're in an electric chair. So it depends on what product you're talking about.
Okay, thank you.
I'd chip in there and say, sorry, I was asked the
Yes.
question about the male egg as well.
Yes, yes.
And would that be safe to eat? And the answer is, at the moment, it isn't a product. If it were a product at some stage, the gene technology and the food safety regulators would be asked that question. What about that waste product, in inverted commas? And the answer is relatively simple. It clearly is inherently safe, the protein that's added. The parent bird and all of the generations above are completely normal and healthy, and they carry that protein inside every cell in their body. And of course, the classic thing, and Alison, I know is well aware of this, that there's maybe a lack of understanding in the general public that when you consume something, when you eat it, your digestive tract breaks it down into individual building blocks, and you're not absorbing the protein. Nonetheless, it's a question that would be asked in terms of, is that protein allergenic in some way? Do people respond poorly to a new protein that's not been in their diet before? So those are questions that have to be asked, but inherently it should be completely safe. In reality, the purpose in the first instance is not to have that protein present. But I've got no objection to it being there. I'd be happy to eat those eggs. Very happy indeed.
Okay. Actually, we here in the US, there is a shortage of eggs. And I was thinking about that because of the H5N1. Prices went up already since last week, and people are hoarding. So it's a bit problem here. Okay. One of the questions would be on which Alison already alluded to, is going to be from Ray Ordonio, is gene-edited plants. There is an intermediate GMO stage because of the random insertion of the transgene into the genome. Is there also a GMO stage for gene-edited animals intended as a non-GMO?
Not in the same way as with plants, because and a lot of it has to do with cell walls. Plants are horrible to work with because of their cell wall, and animals are beautiful to work with because they don't have a cell wall. So we can basically introduce editing reagents using either microinjection or lipofectamine or electroporation, and get actual Cas9 protein bound up with the guide RNA into a cell without needing any plasmid or vector or whatever you want to call it. And so there actually is no intermediary, and it's quite different to plants. And I used to work at Calgene, the Flavor Saver tomato place, so I do know a little bit of what I speak. And so, yeah, it's quite different. And so a lot of the concerns associated with the vector still being present in the plant and all of that is not relevant to animal, and it depends a little bit on your application. So with Mark's application, you probably did use a plasmid of some sort. So there's no blanket answer there, but certainly in our editing we never use any vector or plasmid, and so there's no possibility of having any of that left behind.
Okay, yeah, thank you. And also related to that, this is a question that Julian is going to answer. What are the risks of unintended genetic consequences when applying CRISPR to wild populations, specifically endemic species at risk of extinction?
Yeah, I think in everything there will always be risk. So
Mhm.
in terms of gene editing for our let's take it this way. It's more like in the Philippines, we are still exploring the possibilities, the potentials, and the policies. So in terms of diversity, it's still a very long, tiring. It's a long way to go,
To
to be
go,
quite
yeah.
honest.
Okay.
Um, we're of course we are progressing. We are progressing very well. But unless we know the proper genetics of these animals for the native breeds, the native species, the diversity will be affected. Well, I think Allison was also able to discuss that with the gene editing with the cattle. She was able to show that it's how you use it. If it's just a tool, or if you use it as a founder. So it's very different. You will, because if in, like, for example, in livestock, as she alluded earlier, that there are a lot of breeds, and there in the Philippines, we are the, well, you can say that the native breeds are not, it's not like the commercial ones, because these are very few in numbers. But you need to think about which one are we editing, the commercial ones, or are we editing the native ones? So in terms of diversity, it will differ on how you actually execute the plan. And I think in the Philippines, we have the native, we're trying to actually strengthen the diversity of our native animals. Not
Mm-hmm.
only because gene editing is, but in the Philippines, it's actually very novel. It's very new. We need to protect it first. We need to be able to protect it first, to be able to do modifications, and to be able, it's a very, very long journey.
Okay.
Or the timeline is still very long in terms of the Philippine setup. Yeah. But
Okay.
in every technology there will always be risk. But again, as Alison, I think, have alluded earlier, that should we
Okay.
not do it?
Wonderful.
Yeah.
Yeah. Yes. And Mark has an addition. Do you
Yeah.
have an addition, Mark?
Yeah, it's interesting because, and it was a very good answer, Julianne. We think about this in terms of, I mentioned conservation with bird species because of the technology we have. And I guess there's a number of different ways of looking at it. One, the idea that there may be unintended consequences, which was within the question, when modifying an endemic species that's endangered, you're doing it for a reason. There's a reason it's endangered. If there are other ways of mitigating that threatened state, then
Uh-huh.
use those. If you're using gene technology, there are considerations. One, an unintended consequence. If the species is going to be lost, then that's a
Uh-huh.
risk that is worth taking if you can protect it. The other side of it is one that actually pertains to all the agricultural aspects, maintaining the genetic diversity of the population that you are editing. So it's not sufficient to make one chicken with a marker that lays only females and no males. You need to do that to a large number of animals to maintain genetic diversity for forward breeding. That's something that Alison may have a comment on, but I think it sort of pertains to this issue around endemic species as well. If you've got endemic species that are agricultural, which I think is the focus that Julianne had there, then that's really important too. So you have to consider many things when you're dealing with animal genetics.
Okay, thanks. So now let's go to the question on customer appreciation, or how the consumer appreciation, and would they need to know whether their products, the products they're eating, are gene edited, something like that? And Allison would like to answer that.
Yeah, I think the question was whether they're going to have to be labeled.
Okay.
And, it always sounds kind of innocuous. Oh, just label it. But just think about, okay, so let's just pretend I have a gene edited cow, and I have 500 cows in my herd, and she's one of them. So if I have to keep her milk separate because I need to put a label on it that says she's gene edited to not have horns, for example, let's just say, then that introduces a whole range of costs into the system, because I basically need a separate bulk tank of milk that gets milked with just the gene edited cows, and the rest of the cows go into the other tank. And then I need to keep that separate the whole way through the supply chain. So cheese production, whatever's going on. And we absolutely do that when there's a food safety issue like peanuts. So if you've got a peanut allergy, you need to know it was produced in a peanut-free facility, and it needs to say on the bag if it's got peanuts, right? It's like, because someone could eat that and die. In this case, the cow that has the horns or doesn't have the horns differs by 200 base pairs. There's literally nothing different about any aspect of her milk, meat, nothing. She just doesn't grow horns. Is that something that consumers need to know to such an extent that it's going to basically double the price of the product because you need to keep them all separate? And so I think that, you know, food safety or food labels have historically been kept mandatory for food safety hazards like contains peanuts. If you're interested in a production system, so I only want free range eggs from chickens that have been on green grass, that's typically what's called a value added label, where consumers that want that buy that product, but they pay a price premium because it has to be kept separate in the market. We don't normally put a price premium on just normal conventional milk that tells you something that's unrelated to a health aspect of that product. It's just a production issue. Because people that choose, for example, say I don't want genome edited products, so then, okay, you can go buy organic because they won't allow that in the food supply. You already have that option in the marketplace, but you don't want to make milk for everybody else more expensive who don't actually care whether or not the DNA for the pole allele was in their milk or not. And so I think it's important to say, what is labeling for? And I would argue it should be mandatory for food safety, and I think most people would agree on that. But voluntary product or process-based, like this is how I grew this product, that gets into a niche market where some consumers will care, and that's fine, and they can pay extra for that product, but most consumers don't. And if it's not a food safety concern, mandating labels, especially for something like this where you literally can't tell any difference, is going to be incredibly expensive. And for what public health benefit? I would argue none. And so therefore, I think it should go into the voluntary process-based label speciality market, which is where we put, you know, other stuff like cage-free eggs, for example.
Yeah. Okay. Thank you. We have one question for, where is that? Yeah. This is, I don't know how you would answer this, Mark. Broiler chicken grow very fast and accumulate many heavy and toxic particles in their body. Is there a way out of this problem that GM chicken detoxifying while growing rapidly? How do you tackle this issue arising during GMO development?
Yeah, that's a very interesting question, but I think it's really directed more to the production environment. I'm not certain that we could envisage an animal biotech approach to dealing with those kinds of issues.
Okay.
You know, and it's actually a good case in point, which is that you know, applying animal biotech, you have to find a reason for doing it for which there's no other solution. And then there has to be value in going through that process and determining what the outcome is, as we were just talking about there, whether it's edited or whether it actually involves foreign DNA and that is resident in the product and therefore invokes various regulation, review, etc. So you have to weigh and balance. And I think at the moment, things like avian influenza are a far more pressing problem than something like this. That's not to say that it's, you know, it's invalidating the request in any way, but I think it's something that the industry can look at in many different ways, maybe not animal biotech.
Yeah.
Well,
in the same way. Yes.
I guess I would challenge a little bit the assumption that this is because this would be true whether it's GM or not, but it's basically saying that fast-growing broiler chickens have heavy and toxic particles in their body, and I'm not aware of the data on that. I mean, we eat a lot of chicken, as Mark showed, and we eat increasingly more chicken. And to my knowledge, I'm not aware of any suggestion that consumption of chicken should be limited like it is for fish with mercury. So I guess I would challenge that assumption because I don't think the data supports it.
Okay, yes. And also, there's, I think, two or three questions on the odor of the manure in domestic animals. Would this be possible to control through biotech? Anyone?
Well, it's mostly hydrogen sulfide is what that charming smell is that we all enjoy. And it's just naturally occurring bacteria breaking down the manure into its component pieces. So I guess you could try editing the microbes to not use that pathway. But I guess I'm a bit of a believer that if nature has been selecting microbes for millennium to do a job, whether it's producing methane in the rumen of cows or producing foul-smelling sulfur when it digests manure, you're going to have to produce something that's outcompeting that existing microbe. And nature does a pretty good job optimizing things as a rule. So you'd have to come up with a microbe that's able to not produce hydrogen sulfide that's more efficient and fitter than the existing microbes that exist out there in nature. And yeah. I would not put my hand up for that job myself.
Okay, thank you. There's a question here for Julian. How do you rate the Philippines now, listening to our two experts? Have we already moved fast enough for animal biotechnology, or are we still behind? And well, this is actually a part of your talk as well, so I don't know if you would like to repeat that, but maybe he would like to know more about what's going on in animal biotechnology in the Philippines and what are we expecting perhaps?
Yeah. I think the Philippines, well, it has made a significant progress in biotechnology, of course, especially in agriculture and genomics. But I would say that in terms of growth, it's not as fast as we could hope for. Because there are, well, if you think about it in animal biotechnology, there is still, well, the guidelines is still being considered. It's still on a draft mode. And, another core problem, it's not really a problem, the core challenge that even me I face is funding challenges. So the country is not stagnant, but it needs stronger policies, it needs more investment, and I would say more industry partnerships to accelerate advancement in biotech research and application. I would say that. And I would say more funding.
I think in the developing world, that's not only in the developing world, even in developed countries, that's one of the problems. And as we look at we have a lot of challenges, but we have the tools, we have the technologies to combat this, but the opportunities are there, but the funding or how are we going to solve without funds is difficult. And we know that our scientists are there equipped scientifically. And so, I hope that more funds will be diverted to research globally. Yeah. One of the questions here is on, where is that? How could, because there's a lot of intertwining questions on productivity, safety, and acceptance. So I think this would be a good way of maybe summarizing your thoughts at this point. How do you think would animal biotechnology contribute to all of this? Well, Mark Tizard presented the one on the eggs and on trying to maximize the production of female eggs, and then what other traits are we looking forward so that we can look at the totality of what is in store for the poultry. The same way with the livestock. Already Alison has given us a number of traits that can help us overcome our problem on food sustainability. How do we sustain food and how do we sustain environment? So let us focus on the traits, because some of our people are looking at, do we have vaccines already for swine fever, for H5N1, and for heat resilience, and so on and so forth. So can we start with you, Mark?
Yeah, sure. As you probably know, and I've spoken in many other forums about this, when we set out with having discovered that we could actually modify chickens and edit the genomes and introduce new things in the genomes, we basically sat and looked at what are the things that trouble consumers as well as producers, because we always think firsthand of producers and the problems they face, but ultimately it's what problems do the public have some sort of knowledge and perception of, so that when a product that uses that technology is presented to them, they have a win or a benefit. And so, you know, the three things that I mentioned that we have worked on, we started working with looking at trying to make chickens resistant to bird flu. The nature of the laboratory that I come from is focused on viruses and viral disease. And so our sense was that if we could do something that mitigates the development of avian influenza, that reduces the pool of animals and viruses that expose humans to the potential emergence of a pandemic flu. And so that would be acceptable. We, through the journey, have found that it is most likely going to require more than just an edit, and therefore will be classed as a GM product, or will have some kind of label over it in that respect. Along the way, the use of the marker to track the things that we were doing to tackle the virus presented the opportunity to us to look at the issue of male chick culling. And that has the three benefits of using the marker, but it's a null segregant, not a GM product at the end for the consumer, but involving the potential and the positive application of animal biotechnology to solve problems. And it brings home removing the ethical/welfare problem, improving the carbon footprint, and therefore and also stopping a waste stream and making it higher value, potentially improving sustainability. 3 points that we believe the public would get. And then the final one that we are doing is trying to remove an allergen. Many people are allergic to products in eggs, and we can tackle those allergens and potentially remove them in a way that just uses gene editing. So that was our approach, but we are well aware that there are many other production species that have similar characteristics. And I know obviously one that Alison had spent time working on is another one that people would get. And I think it's thinking about the consumer and what value do they see in it, but there has to be a value for the producer for them to adopt the technology and introduce it. They have to make some return on the investment they make in taking this technology on.
Yes, thank you so much, Mark. And Alison?
Well, I mean, I guess I realize I'm a geneticist, so I'm extremely conflicted in my responses, in that I'm going to always bias towards a genetic explanation. But I would argue that genetics is perhaps one of the most important disciplines associated with sustainability. And so if we can breed animals using whatever methods, I'm not married to gene editing or genetic engineering. Just if we can have productive, disease-resistant, climate adaptable animals that are, get pregnant every year if they're a cow, or they lay an egg every day if they're a chicken, in our production systems, that efficiency and productivity, although it's kind of poo-pooed in the Western world, is actually really important in reducing the environmental footprint of a kilogram of beef or an egg. And I mentioned dairy earlier. A glass of milk today in the United States has about 1/3 of the carbon footprint it did in the 50s, because the cows are 4 times more productive. So if you're interested in the environment, which most most people that live on Earth care. Maybe some people are going to go to Mars, but I'm planning to stay on Earth. And so I really hope that, you know, we can make agriculture as efficient as possible, because then we have agriculture getting produced in food land, and then the rest of the land that's not getting used for agriculture can be used for other purposes, like, you know, in biodiversity and in native species and what have you. If you don't have productive agriculture, because you've got to feed people first, that, like, at the end of the day, I don't really care what's going on in the world. If you've got hungry people, you've got a problem. Once that's taken care of, then you can do the other things that we all value. And I would argue that, you know, efficient, productive agricultural systems enabled through selection for food animals and plants that are optimally suited to that job, which is feeding us, is actually a huge component of sustainability.
Thank you so much, Alison. And Julian, do you have something to share? Any?
I think just a final thought. I think it's a combination of everything. It's selective breeding, gene editing. It's because it's, well, you can say that it's the most sexy way of improvement now, gene editing. And of course, biotechnology. In the Philippines, there's a lot of problems, and especially in livestock and poultry. But then again, the major focus will be on disease resistance, climate adaptability, and of course, high productivity and production. And of course, our major goal will always be to feed the Filipinos. And I think the challenge is to just balance technological advancement with consumer acceptance, always, always. And of course, the regulatory policies that we will have in place in the future.
Yeah. Okay, we have the presentation on regulation, and I don't know. Oh, there's one question here. How do you build public trust in the regulatory of livestock biotech? We have seen, Alison has provided us information on what's going on, and there are many different countries having different regulations, and we're looking at ways of, can we harmonize it? Would it be better to have harmonization so that trade cannot be disrupted, something like that? So any thoughts on what are the other challenges that we have to face for regulation, and how can we make people trust on the regulatory system of animal biotech?
It's a difficult question, and you know, one I think that science communicators are struggling with at the moment in terms of more generally trust in science and well, or perhaps belief in science. And you know, I realize it's been very Australian dominated here tonight, but I actually am living in America. And yeah, it's a frightening time right now to see the misinformation getting spread around technologies like just mRNA vaccines for humans, irrespective of animal, you know, genome editing and stuff. And I think we're having a hard time you know, kind of getting through the noise of the influencers that are putting out misinformation. And it's a challenge. And I don't think that we've been necessarily particularly successful. I mean, certainly we know that if regulations differ dramatically between different countries, it's just going to make everything that much more difficult to get accomplished. So that's an easy answer, but the getting public trust in science and scientists, I think is a much more difficult question, and one that, frankly, I think we're failing at a little bit. And I don't have a solution, because if I did, I'd be employing it and being wildly successful, and I don't think that's been the case. So it's a good question, but I don't have any magic bullets.
Yeah, thank you so much. Do we have any other questions from the FB Live that we have not touched? Labeling is all done. Pricing, do you want to respond to that? Pricing of GMO products compared to conventional? No, I don't think that's an issue.
There's a question here to me that just came in. Is FDA labeling mandatory on genetically engineered products? Yes, it is. Yeah. Do I think that's sensible? No, it's actually not FDA labeling. It's I believe it's USDA is under the control of the Bioengineered Food Labeling Act, which requires labels on products that have ingredients from bioengineered crops. Bioengineered is just another word for genetic engineering. And that's not voluntary, that is mandatory. So we are doing the exact thing that I said was stupid, which is having separate supply chains to cater to GMO versus non-GMO and putting that cost onto consumers that don't care. But yeah, that's what we have, but it is at the current time mandatory. Now that's not true for gene editing when the products do not contain any foreign or transgenic DNA. That's not going to require mandatory labeling. However, there is voluntary labeling through the non-GMO project if people are really concerned. So at the current time, that's how it is. Now, no doubt there'll be efforts to try to make that mandatory, but that's not currently how it is.
Okay.
I can see a
Yes.
question that's come up regarding the flu and bird flu and the idea of raising resistance.
Okay.
And it's a great question because it compares that to the use of antibiotics in animal production, which, you know, is potentially leading to the emergence of antibiotic resistant bacteria. And the question, it really sort of spins around, what if we were to be able to make a bird flu resistant chicken? Would that have a potential downstream effect affecting, well, it's asking here, affecting the human body also? Two parts to that. One is no, it wouldn't affect the human body. Once again, if it was a technique that was involving either a GM component or a gene edit component, that actually doesn't pass to the person that is consuming that product. However, what is very interesting is that there have been a number of publications about techniques that interfere with the replication of avian influenza in chicken cells or in whole chickens that have been changed. And the key thing that has come up there is that we know that viruses are capable of mutating, and mutation is what sits behind the issue of rising antibiotic resistance. Would this happen with viruses? We have to know, and the scientific community that's studying this knows full well, you have to do something that completely blocks the virus and doesn't drive it to change in a way that could become a more troublesome pathogen. So it's a legitimate concern. It's one that the scientific community investigating this is taking on board and has been thinking about for some time, and is working very seriously towards.
Yeah. Okay. One question that came in just now is the objective of GE and gene editing techniques must focus only on for good production, figures and quantity, not the post-production. It might give human health riskier. It might be riskier to human health. What do you think about this, the quality?
I disagree. I mean, you know,
Yeah.
I think you can use the technology to affect different traits, but there is examples where, for example, if you took out allergens, that would affect the food quality, that wouldn't be riskier to human health per se. And I think there are other examples I can think of that would affect meat tenderness, that you know, like a nice Wagyu steak, for example, that would affect product quality that wouldn't be influencing human health. So I don't think you have to look at each example on a case-by-case basis, but I don't think that's generally true.
Okay. I think we are running out of time. And what time is it? It's 10:47 here. It's 11:47 in the Philippines. So before we end, I would like to give our speakers a big round of applause. Thank you very much for all your efforts in coming to the meeting and to this webinar, preparing the slides and also presentation and answering to questions. This is really a very lively discussion, and we're very happy that you were able to impart your knowledge this way and that we were able to induce our participants to raise their questions. So let's give our speakers a big round of applause. Now let's move to the giving of, is that the giving of, no, not the synthesis yet. we would like to go to the giving of the certificates. So this one would be for, we will send you your certificates. So let's start with, what's the first one? Yeah, this is for Allison. So this is for providing her time and expertise as a resource speaker during the 8th International Livestock Biotech Symposium with the theme, Demand-Driven Biotech Innovations Towards a Resilient and Sustainable Animal Industry, held today via Zoom. And while we are showing the certificates, can we just post at the chat box the link to the post-webinar survey, please? Post it at the chat box, and then we are encouraging everyone to respond to the post-webinar survey so that we will know how we fare and where we were able to respond to your needs during this webinar, the knowledge needs that you need, sorry. So LBC, can you share the link to the post-webinar survey? So the next one would be Mark Tizard, and then this is for Dr. Julian Villela. Thank you so much. And then we'll have no, let's have there the post-evaluation form symposium. The post-symposium evaluation form. What is this? Okay, please scan, and we would like to hear from you. We would like to also compare your responses to the pre and now here at the post-symposium evaluation form. Okay, so let's now look at the, we will provide you 15 minutes. Well, after finishing this webinar, we will wait for 15 minutes so that everybody can respond to the evaluation form. So we don't have, we were unlucky to get the person who's going to give the closing remarks, but for synthesis and also for closing remarks, I would like to first thank everyone who have contributed to this successful webinar. We're very happy that our partners are all here, the DA Biotech, DA Bureau of Agricultural Research, DA BPO, Livestock Biotechnology Center, and ISAAA. And also we have participants. We already have we had the maximum of 517 participants, the highest so far. And we are very happy that most of the presentations we will be able to share with you through our websites. And so for synthesis, I really don't need to repeat everything from the beginning, but this is just to remind everyone that animal biotechnology is really a technology that is here, and a lot of improvements, a lot of R&Ds, a lot of products are coming to the market very soon, and that we need to prepare. We need to, in the Philippines, we, as alluded to by Dr. Villela, we're not yet ready, but we are getting ready with our regulation and our capacity building. Our manpower capacities are being developed, and also our infrastructure. And what we need are funding, infrastructure, and manpower capacity. That's really the basic of what the needs are. And these are the things that we need to strengthen first before we go into the regulation. Of course, regulation is important. We have to finish the animal biotech regulation. It has been there for the last three years almost, and we have to be ready. So at this point, the animal biotechnology led by the Livestock Biotech Center, the Fisheries Biotech Center, should be equipped, and our government has already provided some funds, but we need to use these funds diligently and make sure that products will be coming out from there. Our researchers, are not as many as the researchers in many developed countries, but we are slowly developing our manpower through networking, through exchanges, and through continuous education by our scientists. So we will be providing you all of this in our websites, at the ISAAA website and also at the Livestock Biotech Center website. And we hope that you can also encourage your colleagues to look at these presentations and make them aware that animal biotechnology is going to be a thing of the future, not only in the Philippines, but also globally. So with that, thank you so much. And let's give each other a big round of applause. Thank you so much, Mark, Allison, and Dr. Villela, and also Diane for her contributions in the regulation.